And now I'd like to introduce Dr. Harriet Burge. Dr. Burge is widely considered a leading expert in bioaerosols. Dr. Burge helped pioneer the field more than 30 years ago. She developed and ran the aerobiology program at the Harvard School of Public Health for 10 years and has served on 7 National Academy of Science committees on bioaerosols, published over 65 peer-reviewed journal articles, written or edited 4 books, and is a frequent speaker at conferences. Dr. Burge also served on the board of directors of the New England chapter of the Asthma and Allergy Foundation of America. And with that, Dr. Burge, I'll hand the floor over to you.
Thanks, Marvin. Excuse me. Good morning, everyone. We'll start right in. There's a lot in here. We may finish a little early because I haven't taken the slides out, but I'm going to skip a couple that I might not agree with anymore. You never know. I keep reading. Now, this is one of them, and we talk about anecdotal evidence a lot, and a lot of the furor about mold related health effects is anecdotal information that people read off the web or that they hear by word of mouth, and there's no backup data, no data to back any of those things up. And so we try to focus on information that comes from double-blind tests, either double-blind tests either in the laboratory or in the field, and collect data and look at that data and then compare it with the anecdotal data. The anecdotal data is valuable because we get information about where we should go as far as investigations are concerned. But you don't accept that anecdotal data. You just say, okay, this is what we have to do. We have to go and decide this study and this study and this study to document whether or not it happens. Now, with respect to mold, most of the time we look at those anecdotal things and try to follow up on them, and they're proved not correct. Sometimes they are correct, and so it's really a valuable exchange, but you can't leave out the science. As all of you probably know, there are no reliable guidelines for interpreting mold data, indoor mold data, or outdoor for that matter, because you really can't predict it, although we have some very generalized predictions we can make. So whenever you're developing a strategy, you really have to be scientific about it and use critical thinking, think very, very critically about how you're going to do the investigation. And I strongly suggest that you, before you start making a strategy, you develop hypotheses, you develop guesses as to what you think is going on, all the ones you can possibly think of. And then you plan how you would test each of those different hypotheses. Now, can you use one test to document them all? Perhaps, maybe not. Maybe you have to use several. But then you have these questions that you've asked, and you've answered them, and you know how to interpret the data. Now, interpreting the data is always the hard part. You have to establish ahead of time the criteria you're going to use to accept the hypothesis. So if you're looking at indoor-outdoor ratios, what criterion are you going to use and to accept the hypothesis that the indoor air is called contaminated? Are you going to say 1 in 10? I don't. It's a ratio, and you have to decide in advance. I don't recommend doing that, but that's an example of how simple this really is. You know, it's not some complex thing. It's thinking about what you have available to you and deciding whether or not it will help you in your investigation, whether or not it will help you test the hypotheses, or in other words, answer the questions that you've asked about the situation. Now, this is just a list of, you know, we've tried. It isn't that the community of aerobiologists hasn't tried to do indoor standards. The reason we can't is because it varies so widely, and sometimes you can have a lot of mold in the house in the air, and it doesn't mean anything. And other times you'll have a little bit, and it means a lot. And so there's not a way. There's no way. It depends on what kind of mold you have, and what the symptoms are. Now, when the EPA develops a standard, or ASHRAE, or anybody develops a standard, it's usually based on some kind of health effect information. Well, there isn't any health effect information on these mold diseases that, there's no dose response information, there's no real documentation that the diseases can or do occur, and so there's just really impossible to develop. It took me, when I first started in this field, I said, oh yeah, let's say 1,000. Well, that doesn't work. It doesn't work at all. And so these aren't going to ever exist. I've been working on this. Marvin said when you started 30 years, I've been actually working on it for closer to 50 years, and I'm not ready to retire just in case, add that all up and find out how old I am. But at this point, scientifically valid guidelines are not going to exist, and you're going to have to take each investigation as it comes and do the steps that we're going to talk about. Bottom line, you're never, ever going to use sampling data as the only or even the primary information on which to base recommendations. You are going to have done the whole shebang. You're going to have to have done a really thorough investigation. You're going to have to make hypotheses during that investigation about what's wrong. You're going to have to test those hypotheses either by visual methods or however you want to test them, like, is this water damage is coming from such and such. So what you have to do is write down a logical progression as to how the water got where it was. That is the answer to that. That's easy enough. You can do that. If there's a lot of mold in the house, you're going to have to, if you can see a lot of mold in the house, you're going to have to decide whether you have to sample, and if the data that you have already, you've seen the mold, documents that the mold is there. If you see it, it's there. You may have to decide to sample with, for example, tape samples to find out what kind of mold it is. Usually you don't need to know what kind of mold. Usually if it's mold and you know it's mold, then you just go about finding the source of the water and then developing remediation plans and going from there. That can be very simple with no sampling whatsoever. So I'm going to say this right now, if you can avoid collecting data, if you can avoid sampling, do so. It's extremely important. It's extremely difficult to interpret sampling data, and sometimes the data won't support your hypotheses for some reason that has nothing to do with reality, nothing to do with whether or not your hypothesis was correct or not. You have to be careful about that. So really the best thing you can do is to say, there's mold and it's right here. You can see it. I can show you photographs of it, having done a tape sample. And I can say that I don't know whether it hurts you or not. I don't know whether it's of any damage to your health, because you do not ever know that. You never know that. And here's a remediation plan. Here's what we have to do to get rid of this mold and to fix the problem that caused it. Those are the things you're focusing on for indoor investigations. I want to stress that you cannot interpret fungal data with respect to health effects. It can get you into legal difficulties. You're probably, unless you know an awful lot more than I do, you're going to be wrong. I know probably as much or more about than anybody about what mold exposure does to people, not from having, well, from having done research. I've done lots of research in my past lives, but from reading all the literature of all the research that other people have done and evaluating it carefully, and there's just no evidence that mold in houses causes anything other than allergic disease, which it certainly does cause, and infections if there's a severely immunocompromised person in the environment. So those are the two things. Now, none of us want mold in our house. It's kind of smelly and it doesn't look nice. And I agree entirely that that should be removed and the problem should be fixed. I just don't want people to make the mistake of saying, okay, I've been sick, I've had 3 sinus infections this year, that's too many, it must be that mold, because it's not that mold. Once you've had 1 sinus infection, you're probably going to get another one. I mean, you know, that's just how sinus infections are. It doesn't have anything to do with mold. So this last thing, if you, we have done, there have been a lot of studies done trying to connect mold concentrations in houses and health effects. And in fact, we don't see the health effects if we measure the mold concentrations. We see the health effects if we measure indices of dampness. So if the house is damp for some reason, the children in the house may or may not have have a slightly increased probability of having lower respiratory infections, not with mold, but with bacteria or viruses. And so the hypothesis there is something it is damaging their immune systems just enough so they can get these diseases. What that something is, nobody knows. It may be mold, although mold doesn't show up as a correlate with those health effects. So you just really need to remember this, that except in a few cases, yes, that you can say the mold is causing a problem, but mostly causing a health problem, but mostly you cannot say that, and it's not useful to say it to anybody. This is just a little note about those of you who do legal work, who have ended up having to do legal work in your practices. If you have written everything down in a way that is not changeable, like in a bound notebook, and if you make a change in the notebook anywhere, you sign that change and the date you changed it, because that's how you do it in the lab. That's how we do it in the laboratory. You write legibly so that I could, if I came along and had to read your notebook, I could read it and say, oh yeah, this is exactly right, and he did exactly what he said he did, and here's all the evidence that he did it and when he did it. Nobody's going to make up a notebook. I mean, that's ridiculous. Each notebook, well, some of these are just ordinary. Entries in the notebook should be by time and date. You can use a separate notebook if you have a really large study that you're doing, a large investigation, a giant hotel complex or something like that. Just have a unique notebook that's dedicated to that. But in your everyday notebook, write a note saying the data for such and such a study is in notebook number, whatever it is. So here's an overall view of what you're going to do when you do a mold investigation. You're going to develop preliminary hypotheses from the initial or letter contact. This is before you've even seen the place. Somebody calls you and says, I've got a whole building full of people who are complaining about headaches, and I think it must be mold. And then you say to them, well, do you see any mold? Is there any mold there? And they say, oh, no, I think they're all making it up. Well, you can't believe any of those things. There probably is mold there, but I mean, there's always mold. There's always mold everywhere. The health effects probably aren't due to mold, but you don't even know that. You don't know whether they are or are not. So you can't take a chance and say, oh, well, you know, you're probably right, and we'll come and do a big mold investigation. So you develop the hypothesis that in spite of what this guy says, there is mold in the building. That's the first thing, because they're a mold investigator. They're not an investigator of health effects, just a mold investigator. And you can say to him, I can tell you whether or not there's mold in your building, an inappropriate mold, whether mold is actually growing in your building, but I can't tell you anything about the health effects, and neither can anyone else. So that's the first thing you do. And so he says, okay, fine, we'll do that. And you say, okay, you're going to go and say to yourself, there is no mold in this building. That's what the guy said. There's no mold. And then you're going to develop a strategy that will allow you to reliably, with high probability of correctness, say that there is no mold in that building. As you go along, you'll plan and carry out your visual inspection. That's the first thing you always do. And you may, in fact, this visual inspection, you may see mold right away. You may say, oh, yeah, I see that. That's mold. Is that all the mold? That would be the first thing you say. I've been in buildings where there's obviously mold. You can see it on the condensation outside the vents, you know, the air supply vents, which cool off the surfaces, and then the cladistorium grows there. But then you say to yourself, well, if that's happening, is there more mold somewhere else that I can't ignore? And so then you do a more in-depth visual examination and say to yourself, what are all the possibilities for where there might be mold growth in this building, especially with the fact that that mold is growing right there on that ceiling tile. And you develop new hypotheses for all of those. It sounds hard, but you know, you automatically do this. What I've done is actually thought through how I would do an investigation, and it takes minutes. You know, you just say, oh yeah, okay, probably this and maybe that. The only difference is that you're going to write them down, because then when you go back to them, when somebody says, well, why did you think that? You'll have all the data right there in your hands to read. At this point, after the visual inspection, when you get to this point, you can decide if you need to sample. If you do need to sample, you're going to have to decide how you're going to sample, how often, where. It gets into a big can of worms. But but you'll do that if you need to. So the next step is to design a sampling strategy. You conduct the sampling and the analysis, and you interpret the data and you make recommendations. So this is an overall view. So the first step is hypotheses. Now the first set of hypotheses are those that address only the environment. I want you to think in terms of there's environmental mold, and that's all we're concerned about. We're not concerned about whether people are being exposed to it, or we just want to know if there's mold here and why it's here. Those are the two things that you want to know. Is there mold in this crawl space? Is the mold entering the occupied space? You aren't concerned about exposure, you just want to know if it is. That's those two hypotheses. That's two of them. There's millions of others. Hypotheses that address only the environment. Those that address human exposure. Is this group of people being exposed to this agent? Is this mold on this wall leading to human exposure? Those are the questions that you ask. Now, you won't have to ask these very often. On the other hand, in this thing we just said, the story we just said, if you go in there and you find visible mold somewhere, you want to know whether the people in the building are being exposed to it. It's possible that they aren't, that you found the mold, but it's inside walls and places where they're very unlikely to be exposed to it. And so then your hypothesis would be that they're not being exposed, and you'd have to test for that with some of the strategies that we'll talk about a little bit later. But you're not talking about, is this group of people sick because of exposure to this agent? You are not talking about that. Exposure versus dose. Now you know that illness with respect to toxins, for example, which is the hypothesis that the anecdotal people have for how the mold exposure causes headaches and fatigue and all that. They say that the mycotoxins are hurting them. Well, that's very unlikely, but that's what they say. And so, but what do you know? You know, if you're having a toxin that you're being exposed to, dose is the big thing. You have to know how much you're being exposed to, and then how much actually enters your body and circulates around and doesn't get metabolized, or does get metabolized and is excreted. You have to know all of those things, and you don't know any of those things for the molds, none of them. You might be able to document, you can document the potential for exposure. We talked about that. You can document actual exposure sometimes. You can't document the dose of any mold disease agent, and you cannot establish cause and effect relationships without that dose information. The potential for exposure, these are specific hypotheses. Is the mold in this crawl space entering the breathing zone of the occupants? I wrote questions hypotheses because you can state these. You can leave off the is or move it. The mold in this crawl space is entering the breathing zone of the occupants. That would be a hypothesis. Is the mold is a question. I'll just go on with the questions. The Stachybotrys on this wall being released into the breathing zone of the occupants. This is an important one because people will go in and do tape samples of walls and say, oh yeah, there's Stachybotrys all around here and all around here, and then assume that that is getting off the wall and into the breathing zone. It may not be. It may not be. Stachybotrys doesn't release itself from walls very easily unless you actually go there and scrub it. And if you find a whole lot of Stachybotrys in a wall, that means somebody has been disturbing it purposefully or otherwise. And then another one is over time, how much Stachybotrys reaches the breathing zone. We'll mention Stachybotrys a lot because that seems to be one everybody is still concerned about. They call it black mold now and Stachy, which I hate. But it's a lot of the black mold that people talk about isn't Stachybotrys at all. There's lots and lots of different black molds, and even Cladosporium can be mistaken for black mold. It's not black; it's really dark brown. But you know, you don't go into most laymen don't go and look at it that closely. Okay, the negative case. We always, in science, we always address the negative case to avoid bias. So if we think that this is what's going on, we say, okay, I'm going to try to prove that this is not what's going on. I think it is, but let's prove that it's not try to prove that it's not going on. You can never actually prove the negative case. You can only collect data and reach a probability level that's acceptable to you. As far as that negative case is concerned. You can't ever prove it. Testing a negative case requires really carefully constructed strategy if you're going to be able to interpret the data in the end. Bias, by the way, I don't know if I have another slide on bias. No, let's go back. Bias is hard. Bias, everybody is biased. Every single person in the world is biased about something. And there are people who are strongly biased toward mold being a problem in lots of cases. And if you look at those studies very, very carefully, you can see that bias in the structure of the studies. You can actually see it in the structure of the study. Not purposely. Nobody does it on purpose, I hope. But you can see how the study design is working toward getting the answer that they want. And that's very, very hard. So that's what you have to do, is say what answer you want and then try to prove you're wrong. Try to prove yourself wrong. Okay, visual inspection. You've done your visual inspection, and these are the hypotheses. There weren't going to be, these are the possible outcomes or hypotheses. There are neither odors, odors nor mold, one. There are odors but no obvious mold, and there's visible mold in the occupied space. These are the things that you might have found in your, or you might have had hypotheses. There's going to be no mold with the visual inspection, and these are the three hypotheses. Now you've tested them, and here's the answer. You didn't find any. You found odors, but you didn't find any mold, and you found visible mold. Exposure. There's no ongoing exposure from indoor sources. Now you wouldn't say no, but you would say the probability that anybody is being exposed from an indoor source is very low. You could say hidden mold is releasing spores and or volatile organic compounds. We'll talk more about the volatiles in a few minutes. Or visible mold is releasing spores. So these are exposure hypotheses that get the spores and or the VOCs into somebody's breathing zone. Now we're going to go on a side trip here for a few minutes and talk about sampling, because I know I said you're not supposed to sample very often, but if you are going to sample, you have to know how to do it. You have to know what the sampling methods are. And we're going to start talking about individual sampling methods, and I don't want to have to repeat this over and over again. So there's three major kinds of samples: surface samples, and bulk samples, and air samples. Those are the three major types. You can come up with a lot of other names for these, but this is what we're going to do. This is how I'm going to say it. I have these in here because this tells you what kind of analysis. See, you have to know the analysis type actually before what kind of thing you need, what question you're asking. Do you want to know if there are any spores? That's the surface samples. Do you want to know if there are any spores there at all? Well, microscopy will tell you that easily. Do you want to know the names of the spores or whether or not they're alive? You're going to need culture for that. Okay. Microscopy might even tell you the name of the fungus, the surface sample. So this is going to be one of my favorite things to do, you'll see. But it's not quantitative. It's never quantitative. Surface, unless you sample the whole room, which nobody's ever going to do. Bulk samples, same thing, microscopy. Now you can't use, we use polymerase chain reaction, which is a DNA method where you look for specific fungi using a genetic method. And you can use that if you have a big enough sample. You need a bulk sample. The surface samples don't have enough bulk to them to actually do that. But bulk samples do. And you could do this by, for example, if you have some dust, you could collect the dust in a little cassette and collect enough of it so that you can do these, all of these. You can even do chemical tests to document, for example, whether you have endotoxin in there or something of that sort. This is not, neither of these are really quantitative. You can collect a bulk sample, like let's say you're going to collect carpet dust, and you collect it from a square meter of carpet surface. That's fine. You have a bunch of dust, and you know how much dust there is for that square meter, and you can say, okay, I've got this much mold for this square meter. But you really can't. If the carpet fills the whole house, you can't extrapolate from that square meter to the whole house. That's silly. You obviously can't do that. So what you do is say, at least in this spot, there's no mold. And it could be that over there in that spot, there could be some. So it's not, except in large epidemiological studies where a relatively large measure of error, statistical error is allowed, you really can't say that. But you can say, if you're kind of lucky and you hit the right spot in the carpet, you could say, yeah, there's mold probably growing in this particular carpet right in this space. And then that means that probably it's growing elsewhere as well, especially if you don't see any difference between that spot and another spot. But it's really not very quantitative. It's often used, and people tell me how much fungus should be allowed in a square meter of carpet. I don't know. It could be a lot. It could be you live out in the country and the aspergillus comes in every night with your farmer occupants and settles out in the carpet. It doesn't grow, it just settles there. But when you sample the dust, you get masses of it. What does that mean? It doesn't really mean anything. It just means that the dust is part of the dust is aspergillus, and it's not growing. It's just there, and it'll gradually disintegrate. Now, these are relatively easy to design. Air samples are more difficult. You can use air samples. You collect air samples that can be analyzed by almost any method you choose. And most of the air samples that we receive at EMLab are cassette samples, the aerosol or one of those kinds of cassettes. And we use microscopy to analyze them. And with microscopy, we can tell you in general how much mold you have in that sample, and some information on the kinds of things that are there. Are all the spores the same, which would tell you a different story than if all of them were different, and so forth. You can culture air samples. Best to use a culture sampler than a culture air sampler, culture plate sampler. But if you can't, you can actually use a filter cassette, a regular filter cassette, and collect what's in the air and then culture it that way. That's difficult. It's not something you want to do by yourself. If you've never done it, you need to get some expert advice on how to do that. But it's certainly something you can do. You can also do the same thing with that sample. You can look at PCR and chemical methods if you have enough dust in the air sample. You probably need about, well, I don't know, you need a milligram would be lovely. You rarely get that. You can use PCR on less than you can for these other things. Sampling is never the primary step for determining the presence or absence of mold growth. Now remember, what we're talking about in these studies is mold growth in houses. Mold in houses is always going to be there, and you're dealing in all of these studies with a background of mold growth, of not mold growth, but mold having come in and settled on surfaces in houses. It's always, or in large buildings, it's always that way. It comes in on people's hair and clothing, and the dog and the cats and whatever else, and the air, the outside air when you open the window. So we're really not worried about that. We're worried about the mold actually growing in the space. And so air sampling isn't the best test for whether or not mold is growing in the space. The best test is to look for it and look for signs of water damage that could be leading to mold growth. You have to decide if you're going to do sampling of any sort. You have to decide the exact site from which the sample is going to be collected, and you need to label. If possible, you need to photograph the exact site, for example, with tape samples and bulk samples, and label the photo to connect with the sample. So with surface samples, you would do obvious or suspected mold growth, or you might do undisturbed settle dust. We do that quite frequently, and try to look at it microscopically to see if, like 2 weeks ago, there was a lot of penicillium here, and the spores are still in the settled dust, but they really haven't done anything. Bulk samples include vacuum dust, which is commonly used, pieces of material, fabric or construction material that you think might have been contaminated, and then of course water or other liquids can be tested. And with vacuum dust, most people you don't use vacuum cleaner, the house vacuum cleaner or the building vacuum cleaner. The fungus grows in those vacuum cleaners. It grows in everybody's vacuum cleaner. And so you just don't want to do that. You want to use if you are going to use a regular vacuum cleaner, clean it good and then use a new filter bag each time. We usually recommend using some other kind of sampler that's dedicated to your process and that you can change the dust collection method change the dust collection bag each time. There are several out there in literature. I don't have them. I don't have photographs of them right here, but if you need to know them, you can look in the literature, or you can send me a question and put a question in the chat panel, and we'll try to answer that. You'll have to mail it. You'll have to email it to Dave, because I have to look that up, because I don't remember all these things every single minute of my day. Okay, we're talking about vacuum dust, pieces of material, construction material. Now, construction material, let me say this. If you send us a whole square meter of wallboard, what are we going to do with it? The first guess is that what you would like to see is, oh yeah, we'll sample the whole thing. No, I don't think so. What we have to do is either tape sample, which you could have done by yourself, or we have to take the whole piece and wash it off and dilute and culture the whole sample. And we're not going to do that with a whole piece of wallboard. We might take a 10 centimeter square piece out of it, but where are we going to take that piece? So in sending us an entire big sheet of wallboard, or half a sheet, or even a square foot, you are asking us to select the sample site. Which, if you think about it, is not a good idea, because who's going to select the sample site are really excellent analysts. They know exactly what to do, and they have a reasonable idea of how they would collect the samples. But they've never been in the field. Most of them have never been in the field. They don't know what mold. They know what mold looks like. They're all degreed mycologists, but they don't know where to collect the sample. Now, do you want to know what that mold is? You collect a sample of the mold. If you want to know how much mold is on the whole space, and you have 5 different colors, you collect 5 different samples of the mold, 5 different tape samples, for example, and we can tell you them all. And then you write down an estimate of what the area is on that piece of wallboard, and you just send us the tape samples, the little pieces, or if you only have one little place, you can cut it out and we'll do a bulk sample analysis. Water is a little easier because whatever's in the water will distribute itself relatively evenly in the water, and you can shake it up, and you could stir the water up really good before you collect the sample, like in a drip pan. If you have water, you can stir it up good and then collect the sample, and you'll have a pretty good idea of what's in there. Before you do that, you might try to remember that you have to figure out how that whatever happens to be in there is getting out. And I've seen, I was in the Library of Congress at one point, and every time they cleaned the cooling coils, which they did regularly because they had to keep the air clean for the books, not the people, but the books, and it would smell like a fish tank like that, because the cooling coils were supporting the growth of those organisms that cause that smell. And that was really relatively easy to fix. They got rid of all those water washers for air cleaning, but but you have to have, if you have an isolated little bit of water somewhere that is never going to get out, and it isn't going to be important for your investigation. I hope making this halfway clear. It needs to be important. And importance has to be your decision. Is it really important to do this or to collect this sample? There are places and examples where it is important, absolutely important. And there are other places where it's a waste of time because you don't know how to interpret the data. That's the other thing. If you know that you don't know how to interpret the data, think it through. What happens if you get some data? What are you going to do with it? Do you know enough to be able to interpret that piece of data by yourself? You should, because you ask me to interpret it. Well, I'm interpreting it without ever having seen the situation that you're in. I haven't seen the environment. There are too many questions I would have to ask you to give a halfway reasonable suggestion. Now, if you said, "Oh, we've got 100,000 Legionella bacteria in this particular little place in a humidifier, let's say, and is that too much?" I say, "Oh yeah, sure," because in the first place, not cooling tower, humidifiers, like little air humidifiers. In the first place, those put water into the air and put the Legionella into the air, and it's not something you want to do. So yeah, that's too many. Empty the humidifier, or else put it out in the backyard or something, and don't use it. So anyway, we can go on. I have a million anecdotes that I could be telling you, but we don't have time to do that. Air samples are the hardest. There's all these places: outdoor air, indoor air near the entry. A picture of where of how much outdoor air is actually coming through the entry. Indoor air near the mold growth. Indoor air near a potential source before and during aggressive action, and indoor control samples. Now we're going to talk about all these. Wait, maybe not. Let me just go back, because I forget how I've changed this. Outdoor air. Well, outdoor air, you need to collect a sample for outdoor air because it might be a day with 100,000 basidiospores per cubic meter of air, and then you're going to see those indoors, and you're going to say, what is going on here? So you do need to do that if you're going to try to interpret air samples. But you need to also take them with 5 grains of salt, because all those basidiospores maybe weren't there yesterday and none of them got into the house or whatever. There's all sorts of things that can happen with the outdoor air. Maybe it just started raining as you walked in the door and all the ascospores were released, and you had millions of ascospores, but you really only find a few of them indoors, and the ones you find indoors are the ones that you just walked in with on your clothes. So this is why it's hard, because you're dealing with this huge background of the same things that you're looking for. So it's not an easy thing, and it's why I try to avoid air samples. This one I like a lot, and I use this one because it's easy to do, and you can look at the air where you see mold, for example, growing on a wall. You could do a tape sample of the mold and find out the name of the mold, and then you could do air samples specifically looking for that particular mold in the air. And you do a series of air samples, one really close, one out in the middle of a room, and one in the next room, and see if those spores are actually spreading away from that spot. That is a really legitimate way of doing it. And then you easily have a way of interpreting that data, and all you have to do is decide how many spores do I have to see in the other room to say it's important. But we can figure that one out. Even I could figure that one out. Indoor control samples are really, really difficult because, as I have said so many times now, the spores are everywhere. And if you, I had a job once where the spores were, the basement carpeting was filled with a species of aspergillus, and it wasn't visible. There was no odors at all. The carpet was clean as a bell, just really looking clean. And the maid was vacuuming at the time, which was why I managed to find them. But so I took a sample there, and then I went to all the other places in the house I could go, and especially the man's room where he was having severe asthma, and we collected new samples. Well, that aspergillus was all over the house, but it was only growing in the basement. And so to remediate that situation, all you really had to do was get rid of that carpet and clean the basement, and the rest of it would disappear over time very quickly. And in fact, that was what was done. And this is one of those few cases when we could be pretty sure that the man was responding to that aspergillus because he was skin test positive to that particular aspergillus. And when we took all the carpeting out, his asthma got a whole lot better. So that's one of the very rare situations where you can actually say, yeah, in fact, that mold growth was causing a real problem for these people, or for this person. There was only one person in the house who was sick. Okay, and that's how we essentially determined that with this method. Indoor control sample. Oh yeah, if you say, okay, here's my suspect place, and then I'm going to go into the farthest bedroom away that looks perfectly clean and sample. Well, in this case, there was plenty of aspergillus in the bedroom, partly because the downstairs was completely finished and was their recreation room, and they were down there, and then they play and do whatever they're doing, the kids, and then they go upstairs and change their clothes. Well, of course there's going to be aspergillus upstairs. It's very hard to find indoor control samples. And so I usually try to design a strategy that is self-controlling, and that is this potential source before and during aggressive action that I just talked about, where you collect a sample and name that fungus, and then look at how it distributes itself outward. And that way you can, and there's another thing, you could find a whole lot of it right there in the first room. You might find a bunch of it, and then less in the next room you sample, and then a whole bunch of it in the third room. So then you'd have another hypothesis that you had to test is where's that third room stuff coming from, okay? Number of samples, as many as you can afford. Collecting one sample outdoors and one sample indoors, you may be able to use the mold score to do that, to test that, and that helps a little bit. But it's just one little score, and it doesn't really tell you, unless it's dramatic, unless you have 10,000 penicillium spores on the inside and virtually nothing outside. That you can say, well, okay, there's probably something in the space that's releasing these spores, and then you have to go to the trouble of figuring out what. But and you probably could have figured, if there's that many, you probably could have figured that out by looking. But otherwise, it's anywhere, if you get anywhere in the middle of the mold range, it's sort of like, oh, okay, maybe, maybe not. Even if you're at the very bottom, you still don't trust that as your only piece of data. You cannot do that. You can't say, oh yeah, this mold score is such and such, so therefore, or a lot of people just use indoor-outdoor ratios, with which I strongly disagree. Say that the indoor-outdoor ratio is greater than 10. Well, maybe that's a problem, and maybe it's not. Most people would say, yeah, that's a problem. But I've seen cases where it wasn't a problem, with 10 spores indoors and 1 spore outdoors. I mean, there's all sorts of statistical and mathematical things going on there that are actually reality things that can be explained by the mathematics that tell you that you can't really make conclusions based on these data. Now, I say at least duplicates at each site. Now, and it would be nice if you could do that. You often can't. You often have to give up the duplicates in exchange for multiple sites, which I understand that, because most people don't want to pay for these samples. And we do the best we can as far as pricing is concerned. But if it got any cheaper, we would have to lower the quality, and we're not going to do that. Okay, and it's important. The quality is important. You have to have good quality sampling and analysis. You have to have good quality analysis. So you'll have to decide at each place how much you need. One sample. If you're only doing one sample in the building, I'm not going to trust that at all. If you've actually found mold and you've done some of these other sampling approaches, yeah, then I can actually interpret those. How much sample? A bulk sample, you need about 1 gram of dust, or you need a piece of a bulk sample with representative sites. Now, this is a way you can do this. You can send a piece of bulk sample and circle the sites where you're really interested, where you really want to know. In other words, mark the parts you want sampled, because we're not going to test the whole thing. We're going to cut a little piece out of it and throw the rest away. Just keep that in mind. You need at least 5 mL of liquid. If you're going to test for Legionella, for example, you need a whole liter. But if you stir up a reservoir and then collect 5 mL, you're going to find a lot of stuff in there. Air samples, we usually use 75 to 100 liters. Indoors, the same. It's important, if you're going to collect a sample outdoors and a sample or 2 indoors, that you use the same volume in each one, because if you change the volume, the limit of detection changes. And so low quantity things, like for example Stachybotrys, is going to, the number isn't going to be reliable from one sample to the next. You can also use the same for indoor mold, non-aggressive indoor. Well, I said that, indoor mold, non-aggressive. With aggression, if you're going to do a lot of, somebody's going to sit there and scrub the wall while you're doing your sampling, you might want to do a couple of shorter samples, like 2-minute samples or 3-minute samples with these little aerosols, just so you don't overload the slide, because it'll be greater. You'll get a number that says greater than, which may help you. It may be fine, or it would be better to say we got nothing with the indoor non-aggressive, but we got 100 spores per cubic meter with the aggressive. It would be better to be able to say that, or 1,000 or whatever. Just watching time. We'll stop at 10 o'clock for 5 minutes. So microscopy. These are the analytical methods. We're going to tell you what kind of information you get from each analytical method. For microscopy, a sample is placed under a microscope, EEE, for 10 mass and E. At 100, we have a lot of people, edit these, and it's amazing how much 5 people can miss. all the same these things. At 100 to 1,000 X. Now usually we start at 100 and just look over the whole entire slide, and then if it looks reasonably even all the way through, and count the biggest things that we know we can identify at 100X, which aren't very many. But alternaria, for example, you can tell alternaria and epicocoma, some of those big spores. And then I think they go to 400. I'm not sure what EMLab does. I usually use 1,000. The microscope does really well at 1,000, and I think you can see more. I think you can see more, but I think 400 to 600 is okay for the kinds of work that we are doing at this point. And these are the questions you can answer. Is it mold? Is it mold at all? Yeah, but I've collected samples of black stuff on walls, and it turned out to be rubber released from fan belts in air handling units. And people were dying, supposedly, in this building because of all the Stachybotrys that was in the air handling units. And it was rubber. It was little pieces of rubber. Is it mold? You can tell. Are spores being produced? Yes, you can tell that. You can see the spores under the microscope. What kind of mold? You can usually tell genus of, in some cases, you can tell the genus of mold. You can tell the difference between Penicillium or Aspergillus. If you're doing a tape sample, now with an air sample, no, you can't tell. You can only tell in a limited way what kind of mold you have. The spores have to be absolutely identifiable. So, for example, on our list, we do Penicillium/Aspergillus because on those little samplers, we can't tell the difference between those two kinds of spores. Now, if you do a tape sample and we have the spore-bearing structures, we can tell the difference between Aspergillus and Penicillium. And sometimes we can tell you what groups of Penicillium the Aspergillus is in, like Aspergillus fumigatus is really obvious. So we can tell you that if you're doing tape samples. Tape samples are really good. So many of the spores, in fact, probably the majority of spores out there can't be identified. Usually the mass of the spores can be identified. Numerically dominant spore can be identified, and that's almost always Cladosporium. And then next to that can be almost anything. It can be Basidiospores or Ascospores, and then next to that you end up with Penicillium and Aspergillus, and way down at the bottom of the list you end up with maybe 1 or 2 Stachybotrys spores. And you can identify Stachybotrys spores just by looking at them, which is nice. Culture. If you send us a bulk sample, we wash a piece of it, a little piece, and we dilution culture it and tell you what kinds of fungi we found. So that just tells you what kind of fungus. And the reason we do dilution culture is if the piece that we've washed has lots of fungus on it, the culture plate will be overloaded and we won't be able to count and identify things. So this is a very straightforward method. Swab samples are streaked directly onto agar, or they may be washed and then dilution cultured. I like the streaked samples best, but you can certainly do the washing and the dilution culture. Washing swabs, you don't ever get all the spores off, so it's a semi-quantitative method. But it will tell you the kinds of spores that are present, the kinds of fungi that are present. Air samples on culture plates are incubated directly, and they tell you whether or not living mold is present. Oh, shoot. Do that all the time. Living mold is present and what kind of culturable mold is present. And with an actual culture, we can do pretty well on identifying them. We can certainly identify most of the Aspergillus species, a few of the Penicillium species. The Penicillium is difficult because you have to culture it on 5 different culture media, different temperatures and all sorts of stuff in order to actually know what it is. We might be able to put it into a group, one of the Penicillium groups, with less effort. And then some of the other fungal spores that are important, as allergens, for example, don't culture very well. And some of them don't culture at all. So the dominant mold in the sample may not be the one that grows in culture. It may be some weird thing that doesn't grow in culture. I had a case in Florida where the culture plates were blank for a really long time, and then these little white dots appeared all over the plates. That's all that I got. We got these little white dots looking like little crystal things. And I worried and worried over those and cultured them all kinds of things. It turned out to be a marine fungus, a salt-loving fungus that was living in the air handling, in the insulation, in the air handling units, and in the distribution pathways. And it was a rare, not rare marine fungus, but it's something you certainly don't see indoors. And in most of the country, you would never see it. You could see it here if you sample like maybe 30 miles from my house over by the ocean. You could see it, but mostly you don't. So, and that one we almost didn't see it because it didn't like the culture medium we were growing it on. When we subcultured onto salt-based medium, they loved it. It was great. So there are things that are in on culture plates that won't grow. The spores are on there, and they might try to germinate, but they don't grow because you haven't fed them properly. So cultured air samples, you should always underestimate the actual number of spores present because the spores aren't viable. If the spores, if you have a cloud of fungal spores of the same kind, like a cloud of penicillium spores, only a fraction of them are going to be actually viable and culturable. So you never know exactly. Now, if you have a mixed culture, if you have a whole bunch of different fungi, the cultured air samples won't necessarily be less than the total because maybe virtually all of the spores all of the different kinds of spores were actually culturable. So it's not totally straightforward. I've seen cases where the culture looked like it was higher than the spore count. Okay. Remember this, bias the population toward easily cultured fungi. DNA, polymerase chain reaction. I know this is kind of deadly, but people are always asking me these questions, and if we were face to face, which I would much prefer, you would ask me these questions as we went along. What's PCR? Well, you collect the sample, you send it to us, we process the sample to break up the cells and release the DNA. Then the DNA is cloned using polymerase chain reaction, which is a biochemical process where the DNA is replicated over and over and over again. And then once there's enough DNA replicated, then we submit that sample of DNA to electrophoresis, and the resulting bands are compared to standards. This tells you whether or not DNA or possibly RNA of specific fungi is present. And that means that if you have your standards have to include every fungus that you're interested in. You can't say, oh, I'm going to use PCR and I just want to know everything that's there. No, it doesn't work that way. You have to know what you're looking for. So you might say you want Aspergillus fumigatus. That means you're going to get Aspergillus fumigatus and maybe a couple of other fungi, Aspergilli, that cross-react with Aspergillus fumigatus and nothing else. Fungi not included in the standards will be ignored, will be absolutely ignored. It doesn't tell you about viability. It's quantitative, marginally quantitative, and there's no database for comparing concentrations. So I recommend using this now. I think it's a wonderful method, and I think we could develop panels of assays that would tell us a whole lot. And if somebody would collect a big enough database so that we would know we would have a database, and we could really use this. And it's very, very sensitive. It's almost too sensitive for what most of our I'm assuming when I say I'm assuming that we're all mold investigators. I'm not that anymore, but I was for a long, long time. So, pardon me including, we're just all together as one. I'm probably not as good at actually doing a field investigation as many of you, because that's never been my primary job. When I first started years and years and years ago at the University of Michigan, I was doing field investigations in the allergy division looking for potential sources of asthma exposure in patients. And we were focused on the fungi because I was trained in mycology, and that was my job. And we did solve some patient problems in that very small population, and that's how I got started in all this, actually. Okay, we're just going to finish this analysis, and then we'll have a quick break. Gas chromatography, mass spectrometry. These are two different methods, and you use this first, the GC first, and then the MS. And it will give you very specific compounds. If the compounds are within the database, it will tell you what they are. So it's used for testing mold-related volatile organic compounds. There's no point in using this unless the volatiles are what interest you. Mold can be present without measurable VOCs, and this is a big thing to go through. You need a whole bunch of sample to go through. You can use the EPA evacuated canister method if you want to. That works. But you can, well, we'll go on. As far as health effects are concerned, there's no evidence that the mold VOCs cause health effects in the concentrations that they're measurable in houses. Testing for specific mycotoxins. Air samples to estimate exposure. Well, for one thing, you need quite a lot of air sample to actually collect. Unless there's massive concentrations in the air, which there never is, the estimation is not going to work. It just isn't going to work. You're going to have the detection limit is not where you want it to be. You can use it on bulk samples. Whoops. You can use it on bulk samples to estimate source strength. For example, if you had wallboard with Stachybotrys on it, you could squinch up the Stachybotrys and measure how much toxin is being produced right in that spot. Again, the GCMS method probably would allow you to do that, although usually, they ask, if you're going to do it, they ask for a bucket full of sample to test on. It's mostly used in the food industry. And you can get a whole thing of grain, for example, and have it tested for aflatoxin, a whole bucket full of grain. And the assays will do that. But they're not really great for air sampling for houses, for our purposes. Assays are very rarely used, but they are used to detect specific allergens. Now, alternaria, they're always used for these, cockroach, all these, with the line drawn right there, none of which we're really concerned about today, although you might keep your eyes open while you're doing this, because cats and dogs coming in and out of the house, for example, and rats and mice carry mold spores. And then dust mites are one of the major causes of asthma. So if people are complaining of asthma, this is much more likely than the fungi. But we do have one. We have Alternaria. The actual allergen of one of the main allergens of Alternaria, A1. It's Alternaria. Oops. Alternata. We have allergen for that, and immunoassays. They're quite reliable for that, and they require a diluted, a sample like a dust sample, a bulk sample that has been suspended in a liquid and allowed to incubate. To incubate. I'll say that. But for long enough that the allergen can actually be released into the liquid, and then you can test for the presence of Alternaria allergen in the space. Alternaria allergen does cross-react with several different fungi, but that really doesn't matter to you because they all have the same allergen in them. So you won't say, oh, I have this much Alternaria. You'll say, yeah, I've got Alternaria allergen in the air, regardless of where it came from. Okay. I think we will take a 5-minute break. We have almost just after 10, and we'll come back just after 10:05. Okay?
Okay. We'll return in 5 more minutes, everyone. Thank you.
Okay, are you there?
Yeah, you can start when you're ready.
I'm ready. Okay, this is going to be a case study for hypothesis development, and it's not the best in the world, but we might be able to gain some insight from looking at it. So you receive a call from someone who complains of moldy odors, and she wants to buy the house. I've been in a situation where I wanted a house. I walked into it and just loved it, but it smelled moldy, and I needed to know why it was smelling moldy. And so I did want to know what's causing the odors and whether or not they could be corrected. So these are the possibilities before you even go to the house. And I had thought these possibilities through, that there might be visible mold somewhere that we could look for, or they could look for, whatever. And then there could be mold, but we couldn't find any. And there might be not any mold at all. And then there might not be any odors. Now we're talking here about these possibilities that you think of before you go to the house, before you even go. You just talk to the woman. She may be fantasizing the odors. She may have thrown out an orange, and that's what made the odors for the moment. 100 different possibilities, but these are the things you think about before you go. So then you do a visual inspection. You go there and you look everywhere you can. You know the structure of houses. I hope you all do. Where to look in the attic, what water damage looks like. Remember the fact that when water comes in through a roof, it travels around and comes out all sorts of weird places that you would never expect. So watch for that carefully. You look for invisible mold and invisible water damage that you really can't see. If you see water damage, you're going to find mold, at least old mold. If it hasn't been repaired, then you'll find new mold. But look for so-called invisible mold. Where would this be? Behind baseboards, inside walls, all sorts of places you probably know as well as I do. And you can never look at absolutely everything in the house. The one good thing about visual sampling is that you're taking a very large sample, if you think about it. You're taking a sample of everything your eyes can see and your brain can absorb while you're walking around. And if you take notes while you're going, you won't forget any of it. That would be the best thing to do, is to take notes. And then you might look for other sources for volatiles that might be responsible. For example, if they're in the refrigerator, look at the refrigerator. My refrigerator always smells moldy to me, and it's usually some kind of a moldy piece of cheese or something like that. Cheese is made with mold, and it's made with penicillium, and it smells like mold. So, you know, if it's somebody who loves blue cheese and Roquefort and Gorgonzola and all those Stilton, and has cheese sitting around, yeah, that's part of the mold smell. And who knows? If you have a bowl of fruit, the top ones might look wonderful, but the bottom ones might be all moldy. And that happens a lot in my house also. So, and then there might be, there are some volatiles, you know, molds do produce unique volatiles and they smell like mold, but they do also produce other volatiles that are common in the environment, benzene and all sorts of environments. So look for volatile organics stored in the house, especially in the kitchen or the basement, wherever they might store them. And most logical place to look for water damage in a house that looks good is the basement. And let's see what it says next. Oh, these are just general outlines. We're not going to go through this step by step, because I'm assuming that all of you at least know how to do that, know how to look at the structure and decide whether or not it's moldy. And write this down. Maybe you could make a little computer thing on your laptop that has things, and you just have to fill in the spaces. You just have to fill in the spaces. And that would be really a good idea. I think if I were doing that, I would do that nowadays, now that you have laptops or tablet things. And then you get to the rooms. You need to write down all these, the number of rooms and what it's used for, and are there any signs of water or visible mold, and then you need to describe all these things. And why? Because you haven't developed your actual sampling strategy yet, and you need to know, if you are going to sample, how many samples it's going to take, so that you really take to tell what's going on, any. so that when you're done, you can tell the client, yes, in fact, we're going to need to take this many samples and this is how much it's going to cost. You're going to have to do that in the end anyway, so you might as well have it all written down. And she will be so impressed about how organized you are. Basement, you describe the basement. Carpeting is very, very, very important. Paneling is very important. Are there boxes piled along the walls? Is it very, very dusty? Are there signs of water, and does it smell moldy when you first walk in? Now remember, your nose will adjust themselves to the mold very quickly and you won't smell it anymore. So you have to note it just as you're walking in the door. Is there no ventilation down there? That's another thing. Is the basement at all ventilated? So here we have hypotheses from all this visual inspection. You're testing your hypotheses, and these are the possibilities that would come up. There are neither odors or mold, there are odors and hidden mold, or there's visible mold. Now these three things are your hypotheses, and so if you've seen visible mold, then you focus on this one, and you develop a strategy for that. You develop strategies for whichever one of these is the best. It fits your ideas the best, and your inspection. So in your inspection, did you was there any indoor source? Did you find any indoor source at all? Any possible indoor source? And so then you would say there's unlikely to be. You wouldn't say no, but there's unlikely to be ongoing exposure from indoor sources just because you looked everywhere you can possibly look and you haven't found anything. Now they're going to want you to collect an air sample to prove this. This would be a negative case. You'd have to try to prove a negative case. We'll talk about pacifying people after a bit because you can do that, but it's all sort of made up. And I'm a little leery about doing that. You get into a court and the opposing lawyer says, well, why did you do that? Oh, I was pacifying the client. That's the worst thing you can say. Okay. Hidden mold is releasing spores into VOCs. You can't find the mold, but you smell it. And your hypothesis is that it is releasing spores and it is releasing VOCs. And then if you see visible mold, you know it's going to release both spores and VOCs. So with this one, this is the one where you can say that you don't need to sample anymore. What you need to do is go back and find out why you have visible mold and what you can do about it. In this one, if you have a really strong moldy odor, you're going to find, if you have a really strong moldy odor, you're likely to find the source of it. But if you can't find the source, this is the hardest one. You're not going to do this one at all because you're just going to say it's not worth it, or else you're going to have to collect 100 samples. This one, if you think that this is the case, then you have to go back and search harder for this hidden mold. And if you cannot find it, you have to figure out a strategy for deciding how you can document its presence. So you might need to do air sampling in this case. You might need to do some very selective air sampling. And then this one you don't really need to sample for at all. Hypothesis, no odors, no mold. So this is a negative case. You have to decide on the risk of no further effort. If there's no risk of no further effort, if there's no risk, if you don't do anything else and you really think it's negative and you don't do any more and you could go home with a good conscience and sleep well, then that's what you should do. And educate the homeowner on the facts that you've discovered. You've discovered that their house is one of the cleanest and best well-kept, nice, beautiful houses that she's ever seen, and that you couldn't detect any of the odors, and she'll say, oh, well, I can't smell them today. I smelled them yesterday. Well, that's likely to be some local source that she smelled from, a piece of clothing that had become moldy or something like that. Now, the negative case, you could sample in case you missed hidden mold. And if you do, you're gonna sample at electrical outlets and use microscopy electrical outlets on the side of the room where the air is actually coming out. If you have a thing to measure airflow, you would actually do that, and you would want the air to be coming out of the inlet. I don't usually use those wall checks because you could have a very tiny amount of mold and happen to pick it up on that wall check, and then you would be open walls for no reason. And if the spores aren't coming out at all, then you don't need to worry about it. If it's wet in there, that's another story, and you could do a check for water in the walls. That would be excellent. You can do tape samples of undisturbed dust using microscopy. That would tell you if there's a good analyst would be able to say, oh yeah, there's too many of this mold spore in this sample. I'm really surprised to see this many. So you better look for some source of acremonium or some ridiculous mold that the analyst is smart enough to know about, and she can say, he or she can say, yeah, I think this is evidence that you should look harder. That's about as much as you'll get. You can collect indoor-outdoor air samples, which is what most people do, and don't use just a straight indoor-outdoor. Please don't use that unless you know an awful lot about how to adjust for all the variables. If you are going to use indoor-outdoor air samples, you see every single individual fungal type has to be considered independently. So you have to look at alternaria levels indoors and out and what that ratio is. And you have to look at penicillium and aspergillus levels and what that relationship is, and basidiospores and aslos. All these things have to be part of your thing. You can't just say 100 out here and 200 inside. It doesn't work that way. So use the mold score. I don't know of anything else out there that will help you if you're going to do this kind of some relatively simplistic sampling. Okay. So what you found is that hidden mold is absent. Wait, my phone rang, and so now I got mixed up. Here's the hypothesis. We just went through that, and then here's the interpretation of data. If hidden mold is absent, there should be no dominant fungal spores in wall samples or in tape dust samples. Indoor and outdoor fungal population should be similar in quality. And indoor concentrations by spore type should be lower indoors than out. So that would be how you would interpret the samples. The indoor and outdoor fungal populations, and this one are for air sampling, and that's what the mold score does for you. It kind of interprets those and tells you what the probability. It isn't really probability because we're not collecting enough samples to do that, but it gives you a score that along with all your other observations can be useful. Odors but no mold. You found odors, but you haven't found any mold anywhere. You assume the odors are mold related and design sampling strategies to locate the mold, which isn't obvious. And then you use a strategy similar to that for hypothesis 1 that we just talked about. Fortunately, you have these slides. You can look through them, and if you don't understand something, write it on the slide, write it on the PDF that you get, and then ask me a question about it on the internet, and I will try to answer the question. Hidden mold is absent, the same as strategy 1, and you need to consider other sources for odors. Hidden mold is present. Then you would find these are dominant on tape samples, or a single spore type dominates wall samples and both spores and hyphae are present. Now I do this because you can have a lot of mold inside walls. One house I was in had masses of corn smut inside the walls, and I know that corn smut doesn't grow indoors, and so I could just say it's irrelevant. It's blowing in somehow through the cracks from the grass next door, from the field of hay that's next door or something of that sort. But if you have a single spore type, like a penicillium spore type, and both spores and hyphae are present, then you do probably, it's worth going into that wall and seeing what's going on. And then, of course, you're going to be stuck with having to go back and sample all the other electrical outlets or whatever to find out if that's the only place. Or maybe you can hypothetically do it just by what you know about how water moves. Visible mold. There's probably no sampling needed. We have talked about that. You can sample to prove to the occupant or the realtor or the court or whatever that what you see is mold. And so if you think there's any chance of this going to court for any reason whatsoever, like when I was buying my house, there was no chance I was going to go to court about anything. All I wanted to know is I wanted the house and could I get rid of the mold. But if you have to prove it to somebody, then do surface sampling of apparent mold growth, so that when they say, how do you know that was mold growth that you were looking at, you can flash up a photograph of the pictures of the mold that you saw and say, this is such and such. Oh, you can sound so intelligent. It would be great. You could say this, and the analyst, your project manager at EMLab will help you with this. You need to remember that we are committed to helping in this kind of way. If you run into questions of this sort, that's one of our big advantages that we offer. We prefer tape samples in this case, and this would be an excellent approach. No mold. In this case, there was no spores or other fungal elements on the tape sample. Now, we weren't talking about what I was just talking about, legal stuff. What you've done here is tested to make sure that there was no mold. Like in my hospital case where I sampled the black stuff and it turned out to be rubber. That's what this one means. If you have mold, this is what you can use to prove to the occupants and or somebody else that that there is mold there. It only tells you whether or not there was mold at that little specific tape site, and it doesn't tell you the extent. Now, if I were you, I would sample at one or two places to make sure that what you're looking at is mold, and then estimate what your eye is telling you, estimate the surface area, and be sure to mark where you put the tape sample, so that those of us who have to come later can tell what you actually did. So in this case, you got to say no exposure. And if you wanted to prove it, you'd have to do endless indoor-outdoor air sampling to document similar populations. You have to do multiple sampling indoors with windows and doors closed, and you probably will never be able to do this. If you have to, if people are just fussing and fussing at you, just say, okay, it'll cost you. This is what it's going to cost you. And it's a lot. So you can't prove the negative case. What you can do is come up with a probability that the negative case is true, some probability. And in order to satisfy anybody, it's going to have to be a high probability. So that means you have to do a lot of samples. And these are just the means of interpreting those data that you end up with all that. Mold score is low for every single sample, really low. If there's possible exposure, the mold score will be higher, a little bit higher than you expect. And we used to use this 10 times by 10-fold, indoor concentrations higher by 10-fold. I like the 10-fold number because data doesn't become really statistically significant until you get at least 10 hits, preferably more. So you would use these as possibilities for saying, oh, well, okay, I looked and looked and looked and I didn't see any exposure, but I see this, and so I better go back and look again or do more sampling or something. Okay, hidden mold, spores, and VOCs. Design a good air sampling strategy. Spore trap air samples near the suspected hidden sources. And actually, you could do 75 liters, but you might do at least, well, you have to collect lots of samples. That's the bottom line. And then consider doing non-aggressive, aggressive sample pairs. For example, you could collect samples near the hidden sources, like at a why did that happen? Here we go. You could collect aerosol samples near the outlet before and after aggression has occurred. Aggression. Okay, so what you do is you collect the sample and then you pound on the wall right where the sample is and continue to sample. Take a couple of more samples after that. And that might tell you that you do in fact have something inside that wall that can be aerosolized and can be put out into the room. Now, let me say, some of you have abilities to look at things that I don't have. So if you have a scope that you can poke into the wall and look around and see what it looks like in there, fine. That's even better. That's visual inspection of every single little place. Another thing that some people have are microscopes that you can put up against the wall and say, oh yeah, that's aspergillus. Or that certainly is a fungus. Look at all those little spores. And that's great because then you don't need to do any sampling. You've already done it visually. So I have one friend who does that all the time, and I've gone out with him and found all this fungus all over the place that nobody knew was there because you really couldn't see it with your eyes. But when you actually looked at it with this little microscope, long chains of aspergillus spores all over everything. It was wonderful. It was wonderful. I don't know whether it was wonderful or not for the owners, but it was good for a mycologist. Should you sample for fungal VOCs? My answer is no. What you're trying to do is say, yeah, the fungal VOCs are there, therefore the mold is there. And you could do that. You could do that. Mold is sporadic about producing VOCs. It's not going to produce it probably at all when it's dead mold. When it's living and active, it will produce VOCs depending on its current metabolic state. And so you're coming into a whole lot of variables that you won't be able to control. And we do know right now that there's really some pretty good data saying that the VOCs don't cause any health effects that we know of, at least not in the concentrations you get in a house or a building. And this is how the interpretation would go. You'd have all your air samples and you can look at them this way. Hidden mold, unusual spore types, or high concentrations of 1 or 2 spore types on at least one sample. Or, and I didn't put the aggression increases the air sample, increases the concentration of a single spore type. Now, if you have a lot of little spores in there just resting, waiting for the water to come, or not waiting at all, dead, you'll see them when you do this aggression, they'll come out. What you want is to know whether there's colonies growing in there that are making spores. So like you want to know if there's a whole bunch of penicillium in there, and that's what you would look for. If you do find visible mold, this is hypothesis 6, releasing spores and or VOCs, you can assume that exposure is occurring. You can assume some kind of exposure is occurring, and especially, people sitting close to it. I had one way back in the historical times. We had a patient who was really bad, had really bad asthma, and when we pulled out the furniture, there was mold all over behind the furniture, not visible otherwise, and it was all the same mold, all of it, and it was an aspergillus, and she was sitting close enough to it. You didn't see much of it in the air, but you saw a lot of it when you moved that furniture, and she was sitting close to it and having a really bad time. She was in the hospital most of the time. So if you don't see it, you'd have to sample. If you do see it, yes, you can assume, even though you don't get air samples that are positive, you can assume that there probably is some kind of exposure. And in this case, we cleaned the house, had the house cleaned, and fixed the water problem, and she came back home and was fine. So you can design a sampling strategy. In her case, we didn't have to do that. She was very happy with just being able to live in her house. But we could have designed a sampling strategy to document spore release from specific sources. Oh, by the way, we did do one sample. We sampled with a little vacuum pump and a filter cassette the spores so that we can make a skin test extract to tell if she was actually sensitized to that. But, this was an allergy clinic and run by a well-known experimental allergist, and he needed to know that. She didn't need to know that, but he did. Yeah, and we've talked about how you do this, and this is where you would use this method that we talked about way back in the methods part. if you didn't find any spore release from visible growth, and spore types would not be present on the surface, would not be present in the air samples, and this essentially means that under normal circumstances, the spores aren't getting off the wall. It takes quite a lot of energy to get spores off of the wall, even with Penicillium and Aspergillus, and the mold is there and the exposure isn't happening. Do you leave the mold there? I wouldn't say so. I would say recommend that if you could figure out the source, that would be great. I mean, the source of water, that would be great, and you recommend remediation. And because you've designed this nicely, you can say that you can look and say there are some spores right close to the growth but not further away. You can say that if you go in there and scrub the wall, you're going to get exposed, but otherwise it's not being a problem in your house. If you do, you would advise them not to do it without you, because you want to contain the area while you're doing the scrubbing, because then you would have mold all over your house if you didn't contain. And then surface spores abundant all through the house, then you would say there's certainly exposure going on. So the exposure does not equal dose. It does not equal dose. Exposure is not sufficient evidence for cause and effect. Just because you find mold in the air, it isn't the case that that person is being affected by that mold. I've seen this many, many, many times. I've also been followed into buildings by people who do relatively questionable sampling and say, oh, she's wrong, there's ochre toxin in the air and that's what's hurting you. Ridiculous. A lot of these things are just absolutely ridiculous. Just don't get into that trap of saying there's a cause and effect. Just saying, I don't know what's causing your symptoms. It may or may not be this, but I recommend that you do this and this and this and whatever it happens to be that you've decided upon. The data, these are just some recommendations as far as keeping track of what's going on and allowing somebody like me, if you came in to ask me to help you work on interpret a bad case that you've had, or one going into the courts. I always ask for data in Excel format. You know, you can do that on the computer. You can do it directly. You can ask for data in any format you want. I would print out the spreadsheets and include it in your notebook, or you could just download the spreadsheets and put them in your laptop and store them in a separate data book. I'm old, you know, I am getting pretty old here, and I really don't trust computers totally. And I've had enough disasters with them in my long lifetime. The computers admittedly have often been old, but unless you have lots of backup or put them on, you just remember that paper is still the bottom line, sort of. And I know you can all give me excuses why you don't want to use paper, and I agree with you. I think it would be great not to have to use it. And we actually, when I was working at Harvard, we had multiple different files, different places where we stored all the epidemiological data, because we had thousands and thousands of samples, and losing them would cost, I mean, it would be just wasting millions of dollars. So we were really careful. But we did not print them out, obviously, because they were multiple thousands. So yeah, you do it the way you think, the way you are really, really comfortable with. Remembering that if you're sitting in a deposition, you're going to have to be able to pull up the data zippy fast. You're going to say, okay, here's the data. So you figure that out. All of us are at least smart enough for that. Sometimes I wonder about me. So you're going to interpret it. A data interpretation overall. There are visible conditions that have or may lead to mold growth. There are odors likely associated with mold growth. There's visible mold. These are yes/no interpretations based on visual inspection. So you would say, yes, there are, no, there aren't, yes, there are, no, there aren't, and so forth. Bulk samples, what you can actually say, there is or is not mold growth on surfaces. The types of mold present are Alternaria, Cladosporium, et cetera, and so forth. These data are not qualitative, and if you wanted to make them not qualitative, they're not quantitative unless you collect enough samples from an absolutely measured area and extrapolate knowing the distribution of the visible mold. And then if you collect enough samples, document variability, which is a lot, and you relate the sample data to the entire relevant space, which is tricky. So this actually deters some people from even trying to do this. There's sampling data. Non-activity and activity sampling is easy to interpret. Activity concentrations an order of magnitude or greater than non-activity samples, meaning that the non-activity samples are at least 10 or more, 10 counts of an individual taxon or more. If it's less than that, it's really difficult to interpret because the data gets very flaky below about 10. And this isn't 10 per cubic meter. This is 10 counts that were made on your, and there's always a spore count. It's usually adjusted for whatever, for how much of the spore of the surface was analyzed. But you want to look at that spore count and make sure that you've got enough spores in that count to be relevant for interpretation. Spore types in activity samples are similar to those found in bulk surface samples. That's what you want to find out, and then you can say that they're actually getting off the wall. Activity populations are qualitatively similar to indoor rather than outdoor populations. Those are some of the things that how you would interpret the data, and write that in your. I write it in your report. You say we sampled indoors and outdoors. The outdoor samples were much higher than the indoor samples. The kind of fungi were different than the indoor samples, and we think that that any mold that is in your house or came in from outdoors, that kind of thing. And again, I've said this a bunch of times in here, and it's good to say things like this a bunch of times because you get complacent after a while, or maybe you forget. I don't know. I probably would forget. So indoor-outdoor comparisons require the species name of the particles of concern. You can't do total spore indoor-outdoor unless you want to say to somebody, you're getting fewer fungal spore, a lower fungal exposure inside your house than outside. And that house could have mold everywhere and still have a lower concentration inside than outside. So you just don't use inside, outside, okay? Just keep that in mind. Or if the outside is very, very low, like in the wintertime in Michigan, the outside would be almost zero, never completely zero, but pretty close. And the inside would be, 200, I don't know, some number, which is 200 times the outside. That doesn't mean the inside is contaminated. It means that there's always more inside than outside. You've been out walking and accumulating spores on your clothes. You've been in your car. Maybe there's mold in your car. I don't know what y'all do, but there are more spores indoors than out. And you have oranges and apples and peaches and strawberries that get moldy and release spores. So it's always going to be higher indoors than out in a very, very cold climate. So this is an important little statement here. So this is these are a few interpretations of data that were retrieved using the spore trap sampling equipment. And these are the complaint areas and outdoors. Now see here's the raw count, 6, 2, and here's the spores per cubic meter. It's this one. I like to see this one up about 10. Now it often isn't. Whoops. It often isn't. This is the closest. And I'm really surprised at this one. There must have been a lot, and they didn't. That's really surprising to me. I don't know what, I'd have to think about that. How can you have a raw count of this? Oh, I'm sorry, yes you can. Okay, this is very low. This is the one that I get mixed up with these. I don't read them often enough anymore. This is a count clearly higher than this, right? Clearly higher than this for Penicillium aspergillus types. You wouldn't argue with this. You've got Penicillium inside this complaint area too. And you've got it in this non-complaint area. Now, look at this word complaint. That's what this investigator used. They used that word and was looking at complaints and trying to see if the mold spores were causing the complaints. That really isn't the way you should do it. You should look at where the water damage is, where you might have mold growth, and then you wouldn't have had to do this kind of sampling. You would have seen that mold in complaint area too. But a lot of people do this, and that's why I'm going through this. Okay. Cladosporium, this is just how I interpreted this. The cladosporium counts are higher outdoors than indoors. That happens most of the time. Penicillium counts are clearly higher indoors than out in complaint area 2. Now, penicillium, so in this case, the aerosol in complaint area 2 is unlikely to be derived from outdoor air, and the source may be present. Now, that's all you can say. You cannot say, oh yeah, this is causing these people's complaints. Don't ever say that. It's just that there probably is a source. I say maybe, in this case it's so high, there's probably a source of some sort. Now it may be, again, the moldy orange case, it may be that somebody had a moldy orange in their drawer and decided to throw it out on that particular day, and you sampled the air right, closely after that and got all those spores. I don't know. But, you could ask about that. And especially if you can't find any source in this particular area, you would ask about that. The small number of samples, if you only have 1 or 2 samples, you can't say, that's why I said maybe, because you can't say there is a source or there's likely to be a source. You have to say maybe because of what I said, because of the orange. It says non-viable methodology. What that really means is just spore counts. It means that it could be viable. They could be viable. They're just, we don't test for viability, so they're just total. It's a total thing. I have to think of a better word than that. So here, complaint area 1, of the ones, let's look at the ones that we actually have a high enough count on to make it worthwhile. So ascospores are relatively high in complaint area 1 and the same in complaint area 2 and same in the non-complaint area. And I can't see the results over here. You probably can because I've got this control panel there. Okay, here, I'll do this. Okay, so, and look at how many we have outside. So these are obviously they're all the same and they're all coming in from outside. Basidiospores, the same thing. Look at this many basidiospores outside. It was probably raining on this particular day. Cladosporium is still higher outside. And all of these, Penicillium aspergillus, now these are in low concentrations. Outdoors is higher across the board than indoors, and so the penicillin, there's no problem in any of these complaint areas on this particular sample. Nothing. There's nothing here that you can point to and say, oh, this is bad. Now, what if you were using totals? 8,000 versus 1,000, 1,000, 1,000. These are really high numbers, and you think, oh, well, the ACGIH years ago said 1,000 was too many. Well, okay, is this a problem? No, because you look at the ratio, and it's about, I don't know, 0.4 or something like that. So less than an order of magnitude difference, consistently lower indoors, and no difference in exposure. Well, the one thing is that these are thousands of species. The taxa could differ indoors than out. You could have Merullius lacrimans, which is a wood rotting fungus, indoors, and a whole bunch of other kinds of things outdoors, mushroom spores outdoors, and you would never know it. And the same with the ascospores. But in general, this works. This method that we've talked about at this point works. Okay, this is another set of samples from the same place, it looks like. I'll have to change these ones. Alternaria. This is the odd one here. You have quite a bit of alternaria in complaint area 2. And I don't know what complaint area 2 is. You can't count the Cladosporium because it's way, and you can't count any of these because they're way low, and it's pretty high outside. And then we get to this one, complaint area 1, very high, complaint area 2, very high, and outside nothing. So this is how you would interpret this. Now remember, what I'm doing here is going through samples that have been done the way I don't recommend doing them, which is to get one sample, let's see, 3 samples indoors and 1 sample outdoors. And you could do that, but and people do, and in this case it was helpful because you can find the places where the spore counts are out of line. But number one, you don't know really where the growth is, and that's what you need to find out. You don't really care whether or not the spores are higher here or there or the other place. What you care about is finding the mold and getting rid of it. That's the bottom line. And in this case, using this kind of air sampling, you don't find that. You don't know. It's helpful air sampling if you know you're going to go to court. You can say, here's another little piece of evidence. On the other hand, it's possible that your evidence will contradict what you've been saying all along. So you have to be really kind of careful about that. Okay, this is cultural data. We do use culture sometimes, and the main point of this is that, and again, this is the same complaint area one, blah, blah, blah. And these counts are really low, and as they usually are with culture, but we did find, and now penicillium is a spore that grows very easily in culture, so this cultural data goes along with the spore trap data that we had. And in this case, we know it's penicillium, it's not aspergillus, and so we can say that, yeah, there's a possible source here, and let's go look for it. Or we can say this documents the fact, that's the other thing you could say, this documents the fact that in this space, there's exposure, some kind of exposure to the spores. Whether or not it's causing symptoms is a whole other story. We don't know. Okay. So again, no aspergillus at all, and all this. Now, what is this? Non-sporulating fungi actually turn out mostly to be either basidiospores or ascospores that have germinated and grown a little bit. And they've obviously come from outdoors in this particular case. So the purpose for this one is that when you try to interpret these and you see this non-sporulating fungi, you can't interpret this whole thing as a whole. I'll say that again, because these numbers are not an order of magnitude difference, none of them. So this is twice this one, and that's all. So none of these are helpful as far as interpreting what's going on in this space. What is interesting is the fact that you have these non-sporulating things, and that the cladosporium isn't growing inside, even though you have these numbers, it's not growing inside. Okay. This one has some Aspergillus versicolor. By the way, Aspergillus versicolor is by far the most common Aspergillus that we find indoors. It's most common. And in this case, it isn't there, at least not in any amount. And neither is anything else. The cladosporium is the only thing that's in here. This is a slightly elevated at almost 10, but this is way, way, way over. So you'd look at that number only. The only thing of relevance is, the only thing that really is statistically important is this one, and it's one that we don't really care about. Okay. I don't know how much more I've got, but I'm going to go on for just a little bit more. I've mentioned the mold score a bunch of times. I'm just going to show you a couple of reasons why we like it. It's just how we did the data all over the country and distributed throughout the year. So we did an agreement ratio, which I've never really, I guess I do understand it when they explain it to me, but it goes right out of my head. But, you want an R value to be up in the 6 or 7, 0.6 or 0.7, even 1 would be perfect. But this is almost nothing. You can see by the line, it just doesn't matter. These little dots are levels, concentrations, indoors and outdoors. I'm sorry, the agreement ratio. This is an agreement ratio, and this is the experts. These are experts, and you can't. These are scattered all over the place. So the experts and the experts are not interpreting these as being. I'm sorry. I'm tired. I'm not tired. I'm just old. So what we did is have the experts decide whether or not these were in each one of these little samples was represented a bad situation or a good situation with the agreement ratio. First we had the experts do it and assign a score, and then we did the agreement ratio and plotted the experts score between 0 and 100 against the agreement ratio. Okay, that makes it a little better, a little more clear. And then we calculated these little things for the R-squares. So no relationship, and they'll all be like this. The experts score plotted against a Spearman rank correlation for the same data, and you can see no correlation. This is a converted indoor-outdoor, and this is a case where the outdoor ratio is you have to have at least X number of spores before you can use this. That's how they did it. It's you know, if you have 2 or 3 spores, you can't use it. You have to have more spores. It has to be almost an order of magnitude difference. So this one is a little better, you'd think. I think it's a little better. You can see the R-squares a little better, but not enough better. It still doesn't look good when you just look at this relationship. And then here's the mold score. Now, admittedly, the mold score was designed by the experts, but we had a lot of disagreements about it, and so it's a consensus mold score. And we used the experts' interpretation of each data sheet. We had 100 data sheets. We had the experts classify each data sheet from no problem to a high problem, to lots of problems or a high probability of problems, and then use the mold score to calculate the mold score. Now, the mold score now goes from 0 to 300, basically because people were saying this was a 100% chance, and we didn't want it used that way because it isn't calculated to do that. It's just an indicator number for you that it helps you. And the way the mold score was derived was to take, let me see what comes next. Yeah, to take every single one of these and calculate a number, an indoor-outdoor number for all of these, and then we weighted some more than others, because some of these will never grow indoors no matter what, and a bunch of other calculations. I wasn't involved. This is a statistical problem that was not really a statistical thing, but it was an algorithm that one of our really good staff members developed that did all this. And what we were doing, he developed it, and he said, "Could you tell me whether or not this works?" And this last slide says, yes, in fact, it does work. It reasonably accurately interprets, or gives a reasonably accurate guess about how I might interpret it, or Janet Gallup might interpret it, or the other 4 people on our little committee, which were all experienced aerobiologists who've done lots of investigations and interpreted lots of spore sheets, how we would have interpreted that data. So this was just getting our input. All this does is get our input, this little group of experienced aerobiologists, our input into your data. And that's all we meant it for. It's just a suggestion. Here is how we would have addressed just this. Now, is it right or wrong? I don't know. We don't see the conditions or anything else that you see, and but it does work better than the other indoor-outdoor ratios that were being used before we developed this one. So in this particular one, we have spores indoors and spores outdoors, and the only indoor one that's high is PennAsp right here, but the outdoor is higher even than that. So none of the indoor counts are higher than the outdoor count. See that? So this one we would have interpreted outdoor source, outdoor, not different, and this data does not suggest an indoor source. And here's the agreement ratio. This indicates an outdoor. The maximum indoor-outdoor ratio is 1.9, which some people might interpret as a problem, but it really isn't. That's too close. And the mold score is 17, which is really low. And the expert average was 11, which is really low. So remember, this is 1 to 100, 1 to 100 at this point, and this is 1 to 100. So the mold score gave a tiny bit higher, but not much. So this one, the mold score worked really well. Now we have this one where the outdoor ASCO scores were really high, and this is where some of the tricky parts of this mold score analysis comes in about how you interpret that many ASCO scores, how you put it into the equation, or do you at all? And the same with the Basidio scores. This is where, in this case, we have a number that's reasonably high. And so we need to know whether the mold score will pick this up and whether any of these other things contribute, and probably they don't, because this is the only one that's high enough to be actually statistically relevant. Okay, an indoor source is possible. Penicillium and aspergillus are 3 times higher indoors than out, not an order of magnitude, but 3 times, which is not quite what we want, but close. And you need to know whether the indoor and outdoor spores are similar before making a decision. Is the PennAsp, are the PennAsps the same outdoors as they are indoors? Here's the agreement ratio. Outdoors, maximum indoors would say yes, there is an indoor source. The mold score would say yes, there is an indoor source, and the average would say yes, there is an indoor source, and that's based on these numbers right here. Just going to go through a couple more of these, and then I'll quit doing this. When you see this number, you think, oh my God, I've got Basidio spores in the house, but look at the difference. So let's go down here and see what the mold score says. Here's another possibility. This is higher than the outdoors by quite a lot. And then we have Stachybotrys. Now, the committee argued a lot about Stachybotrys and whether or not 8 Stachybotrys spores would constitute a problem. And I don't know whether it does or not, but I was one of the ones in favor of not considering these tiny amounts of Stachybotrys to be important, in spite of the fact we didn't see any outdoors. On the other hand, I'm open to suggestions. 4 times lower, so this is the Basidio spores are outdoors. The Basidio spores are outdoors. PennAsp spore concentrations are more than an order of magnitude indoors than out, indicating a likely indoor source. So I would expect the mold score to be high just based on that. The agreement ratio is still right in the middle somewhere. Spearman says there is an indoor source. Indoor-outdoor ratio says there is an indoor source. This is very high. The mold score is 100%, and this would be 100 if it weren't for me, probably, because I was arguing about this Stachybotrys, this 8 little Stachybotrys spores. But this one you can say there's an indoor source based on this number right here. And this one, this is really low, a low number. And this is one where it's hard because there's a very low number outdoors, very low. And then this is just slightly elevated. So let's see. Indoor concentrations is an order of magnitude higher than the outdoors, but low concentrations make this comparison really, really risky. And if there is an indoor source, it's small. Agreement ratio the same. Outdoor source for Spearman's. Indoor source, absolutely an indoor source for the indoor-outdoor ratio. which is used a lot, and the mold score indicates not an indoor source. And the expert average is about 50-50. So the mold score is saying that these counts are really low and that it's not anything that you need to really worry about. On the other hand, based on a relatively high number here, you might look and see if you could find aspergillus, a source of aspergillus, Parathorium aspergillus in the space. Oh, yes, I've got a lot of these. So this one, by the way, this one we essentially eliminate because it doesn't grow. These don't grow inside. These don't grow inside. These generally don't, and these generally don't. So those we don't usually look at those. Cladosporium isn't important. Here again, we have Penicillium aspergillus, which is a relatively low level. Well, it's almost 1,000. That's something that I would probably say was a problem. 4 times higher indoors than out. And in this case, 4 times is a little higher than 2 times or 3 times, but it's not 10 times, which is what we look. But I would suggest a thorough visual exam. And again, the expert average is just the same as it was in the previous one, and the mold score is a little higher. And this one is lower. I don't know why. I never know what to do with that. This is an outside source and an outside source. So this one is an equivocal one, but with a mold score of 63, it's greater than 50. And remember that it would be 150 now, but that would be the minimum. That would be the middle of the scale. But again, you would go back and look just to make sure you haven't missed anything in this particular area. Okay, so this can be a sanity check in some situations, and that is what we ask you to do. This is an indication of the probability, more or less, of indoor fungal growth. It's meant to be used only in conjunction with a visual inspection by a qualified inspector, a qualified person who knows how to develop hypotheses and who knows how to develop sampling strategies. It certainly can help in large epidemiological studies. I think it would be very valuable there. But I think people complain that we even do this, that say we're taking over your job of trying to interpret data. We're not. We're just giving you our opinion of what it might be, and you make up your own minds based on what you've seen in the space. And if the mold score says you don't have anything in the space and you know you do, then you just ignore the mold score. You say, well, that's their opinion. They haven't seen the space. I'm going to look at my own data. That's all you have to do. That's all it takes. Okay, a couple of minutes of walking around so that my knees don't freeze, and I'll be right back in about 5 minutes, less than 5 minutes.
Okay, we'll take a quick break and return in 5 minutes.
Okay, I'm back.
Great, you can start when you're ready.
Yep. Okay. Now this is another thing that EMLab has developed that would help, at least help you understand the variability in outdoor sports. So that if you're going to try to compare indoors and outdoors, and the reason we do this is that the majority of people do it this way. And we'd like to help those who do. I mean, in other words, my opinion is that you need to do a good visual examination and just like we talked to this whole time, making the hypotheses and the strategies. That is going to be really, really defensible. And you're going to feel good about it. Your client's going to feel good about it. The whole thing. Well, I don't care about lawyers, but anyway. So, but those people who are using this other method, it complicates the field a lot. So we've actually done the mold score to help with that, and then this outdoor air study to help. And I think understanding this variability is really important. And I'd like to do the same thing with indoor spore populations. We haven't done it yet. It's a little more difficult because indoors might be the same in Florida and in Arizona because of mold growth, but I need more data. I would need more data. This is what the sunny day outdoor air on a sunny day looks like. And this is the background on a sunny day of what you're dealing with. And a lot of these grow indoors. This grows indoors. This probably doesn't. And then there's all sorts of little teeny things in here that you can't see. Boom, 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 boom. Those are all here. There's a pink one that you can see. These are all, this is something, the spore. So anyway, this is a lot of allergens in here, a lot of spores that can grow indoors. There's probably penasp growing in here as well. And this is what it looks like when it just starts to rain. You might have that one we just had. It might look like, this, and then the clouds roll in and it just starts to rain and you get this. Now, what is this? This doesn't look like much of anything. But here is a puffball spore. And what happens when it rains is that the rain splashes fungi that are on surfaces into the air, and you see them. That's the only time you see them is when they're splashed into the air. They don't really, they aren't consistently present in the air. And then this is after it's been, now this one is probably a splash. This is the powdery mildew, it's probably splashed. But the rest of these, all these little things, are ascospores that the rain has hit the little spore-bearing structures and swollen up with all the polysaccharides that are in there and squirted out all these ascospores into the air. So within minutes of the beginning of rain, you end up with this. So you can arrive at a house and find out that there's is or isn't mold inside, but when you do the air sampling, you don't find anything. But when you do the out sampling, this is what you see. And we saw some of that in those data sheets that we had. And it could very well be that this is exactly what happened. Unfortunately, we don't know exactly which ascospore these are, but they're so cute. I'm a mycologist because I think that mold is beautiful. So with limited outdoor sampling, unless you can do a whole bunch of outdoor sampling, you need a database to evaluate the representativeness of the data. This is the mold score. We have many, many more samples now. We could develop this. I'll have to ask about whether it's continually advanced, continually adjusted, but that's neither here nor there right now. So in addition to helping with indoor air investigations, it also is a good, research tool for us. So by state, this is how it started, by state, by taxon. By state, by taxon. So you have Arizona, Kaleidosporium for the whole year, averaged out for the whole year. And it's interesting because if you look, you think of Arizona as not having much, but look at this, these outliers way up in the hundreds of thousands. Obviously, most of them are down here. Arizona, penasp, chronological. Why did we use Arizona? We had a lot of samples. There's a lot of others. We have them all, now we have it all zip code related, and it's much easier to deal with. But again, Arizona isn't the absolute non-moldy place that you think it should be. It certainly has days in the middle of the year. The project numbers are by chronological. We have days that are high. This is California, and we have very rare days that are high. Nothing, virtually nothing, all year long. Isn't that interesting with respect to alternating? And this is by month, January, February, March, April. This is, at least in the springtime, we don't have that much. We may in the summertime have more. This is what a mold range data sheet would look like. So this is outdoors in the state of California in August. And you get 40 spores per cubic meter, and you look along here and say, is this normal for August in California? And 40 is the median, so it needs to be close to 40, and it is. By location in California, it should be around, it should be around 13. Oops, I'm sorry, around 27. And it is a little higher than 27 for California, but not much higher. It's probably not statistically speaking. Very common. This tells you how common that spore is in the outdoor air. Very, very common. Look at this one. Very, very common. Okay. And this one, very, very common. Those are and this one, the cedar spores, very common. We have these separated by the way, able to grow indoors, seldom found growing indoors. You should never find rusts growing indoors unless you have the right plants. Okay, I'm going to go on because we can do. This is data by state and by month, January in California, and then we're looking for data by climate areas. California is divided into 8 climate regions. It may be different now, but when I did it, it was this way. And it's calculated based on the database for the region in which the sample is collected. And the samples are groupings of zip codes, and this allows more accurate data than, say, California, because San Diego's aerosol is very different, for example, from San Francisco's. So this is the number of, this is more specific to the conditions where you sample. So this is a state report, and these are all regional reports. And the reason of these is that, number one, we don't have enough samples, for example, in Montana, to do a valid comparison. And the climate in these places. Now, this one I really wonder about. I have to look at that one and see what they're doing with that. But some of these are logical. Most of these are logical areas. So if you sample in Maine, you're getting the results from this little area. If you sample in New York, you're getting just New York samples. And you can see the invalidity of some of this, because actually this part, all this part right here up would be the same, but this part isn't really. So you're adding these two, you have these two overlapping, but we're just doing the best we can for the moment. And it's nothing that you need to say. You need to learn from it and not say, well, we'll show you. I'll show you some examples. The value of this process is you know, that was the end. Actually, that's interesting. I did have samples. Let me go back to this just a little bit. This right now is really quite variable, and I have to add in here a couple of sample reports, because I don't know why they're not here. They should be here. But anyway, a couple of sample reports, because what the reports do, in fact, I had one, this sample report. Now it goes, instead of having it like this, you would have for your region, typical for your date and region, all at the same time, all in one number. And then this would be, instead of this, you'd have this all together, all in one, and it would be not August, it would be August and California and your region of California, all put together into one number that goes here. And so then you could look at, for your region, what the outdoor data in August in your area is. And that can be very helpful. You can see what the range is, low and high, and you can say to yourself, oh, well, my count outdoors are really low right now. So if I see a whole bunch inside for some reason, then they're probably not coming in from outside. If it's way up here for some reason, you can say, oh, well, I have these alternaria spores indoors. These aren't indoors, these are. I have alternaria spores indoors. They're probably, it is true. The outdoor data is probably right, and well, it's high. Our outdoor data for this particular month is really high compared to what it should be at this particular month. I'm sorry, but that's not clear. I have to make a better slide for this one, and I will if any of you decide to come back and listen again. Or you can see the new data when you get it. If you ask for the mold range, which is an extended outdoor comparison, it actually compares your data, which is this, with outdoor data in a very nice way. Okay, we are actually a little bit early, Marvin, so we can do some questions if you like.
Okay, great. So we do have some questions in the queue, and I'll get to the first one here. The question is, when trying to determine exposure and no spores are found airborne, yet are in the wall, I'm not sure what they mean by that, yet not in the wall, how does one show that there's exposure to MVOCs from fungal growth? Does that make sense?
Yeah, I understand what they're saying.
Okay.
There might be exposure. How do you tell by odor? If you can smell it, you know what it is. And you're saying that there is or is not mold. If they're saying that there is mold in the wall, you haven't detected mold in the wall yet. And so then if you found like you could smell the mold coming from that wall, you would actually look in the wall and find out what's going on in there. It doesn't mean that because something's going on that the spores are getting out into the air. And I think this meant that the air samples are really low, and yet maybe you could smell the air and the VOCs are coming out, but not getting spores coming out. And that's what often happens. So then at that point, I would open the wall and look at just make sure that there's no mold or that there is mold. Because if there is water and mold, actively growing mold, then that's damaging the house. May not be exposing anybody to anything, but it is doing damage to the house. Could you collect the MVOCs? Yeah, you could, but if it smells like mold, it probably is mold. I mean, and drilling a hole in a wall and looking around in there isn't going to hurt anything. You can always fix a wallboard. That's not any big deal. So that's what now if you're in my house, it would be a little harder, but I don't have mold. I've got plaster. Anyway, so yeah, I think that's the best answer. By the way, right here at the start, if I give you an answer that isn't what you need, put it in a question to Dave, and he will send it to me. The reason he gets them first is that he tries to put them together in groups so that I can answer groups at a time. It makes it a little easier for me. But anyway, send it on to Dave, and I will better understand your question. That would be the bottom line. Okay? Next.
Great. And a quick update for everyone. Dave's email address is in your chat panel. So if you want to reference his email there, I included it there for you. Okay, so the next question, what is culture samples used for?
They're used, for example, I have a presentation I'm going to do at AIHCE, American Industrial Hygiene Association meeting next month. And it has to do with Aspergillus fumigatus and the potential for exposure to immunocompromised patients, for example. Or any you don't have to do it in hospitals, you do it in houses, anywhere you want. But if you're going to move a transplant patient home from a hospital, from isolation, it would really be nice to know whether you have any Aspergillus fumigatus growing in your house, because that is the most potent of the fungal infectious agent, of pathogens, opportunistic pathogens. So you use the culture sample there because you need to know the name of the Aspergillus, and you need to know how much of it is in the air and whether there's any in the air. And this goes along with really, really, really good visual inspection. Because actually there are other fungi that can infect immunocompromised people. But I would include a culturable air sample just for that reason, just to look for Aspergillus fumigatus. In hospitals, it's often used for a variety of reasons. If you need to know viability, or if you want to know the name of a particular fungus, you would want to use culture. There are many fungi out there that we can't identify on the spore. We don't even see some of them and some of those would grow in culture that, you know, if you had an equivocal case. It's rarely used. It's not used a lot. The people that use it are generally either in hospitals or they have been in the field for a long, long time. When I started, we only used culture because we didn't have these little portable things that we could use. Now it's very easy to do a spore trap sample, so that's part of it. So the bottom line is, if you want to know the name of a living fungus, you need to do culture sampling. Okay?
Okay, great. Next question. Why would you capture a water sample?
Well, there are fungi in water. There really are, and they produce odors. They're unpleasant. Usually the water samples are from humidifiers, the drip pans of humidifiers where you can find all sorts of horrible things. Rhodotorula is a fungus that is actually a yeast form fungus that's commonly found in humidifiers, and there have been cases of hypersensitivity pneumonitis related to Rhodotorula in humidifier water, where the water is sprayed into the air. And you know, I don't generally collect water samples for fungi. I really don't. But again, in hospitals, Aspergillus fumigatus has been found in tap water samples. So somehow it's growing in the orifice there probably, and in a warm spot, and would come out with the tap water. I was really surprised when I heard that, but apparently it does happen. So that was why, those are the reasons why, and in that case you would use culture sampling to find out what was in there. And it you know, likely they're going to be negative, but it depends on how much risk there is associated with not collecting the sample. That's what you have to think about in those cases. A normal, ordinary, a normal person, you, most of you, all of you maybe, would not be affected by Aspergillus fumigatus. I mean, it just wouldn't affect you in any way. And so you don't need to worry about it for most of the time. Okay.
Okay, next question. Would you please comment on people who are using ATP portable testers to determine the presence of mold?
Well, ATP is in people as well. It's in every living, at least every higher of the living creatures. And I don't know where the ATP comes from. It could come from mold. It could come from all sorts of things. They're talking, and it may be that it would show up in bacteria as well, but it may be that it is detecting some kind of mold growth, and that's fine, except that there are cheaper ways to do it. I mean, you don't need, if you use an ATP sample, I guess that means that you aren't seeing anything, and so you want to know if there's something there that you can't see. And I suppose it's useful for that. I just don't know. I don't use it. I'm skeptical about it because ATP is universal. You know, dog spit has ATP in it. Not that your dogs spit on your walls, but mice might. But I just don't recommend it because I don't know how to recommend it. I know there are people doing that, but I think most of you could detect mold that's caused by water damage of some sort. I think you could detect it with your eyes or with a little microscope, which would be a lot cheaper. One microscope and you use it for the rest of your life. If you need to see the little teensy things, if you can't see them, if you have a wall that tests positive for ATP and you do all the things I've said, tape samples, microscope looking at it, and it's negative, you don't see anything, then I think the ATP is wrong. Likewise, if you don't see ATP and you do see those mold things, then the ATP is still wrong. I don't know that anybody's actually done that. Done that test, that comparison. I think would be really interesting to do, is to collect 10 ATP samples from various spots in a house, and then collect, and then have a good investigator go back and look at what they can see without using ATP. I'll have to look into ATP a little bit more, but I just am very dubious about it. But then I'm old-fashioned. Maybe I'm wrong. I'll write an email white paper, an EMLab white paper if I'm wrong, and explain that I was wrong. This is how you can use it.
Okay. Next question: how does one interpret wall cavity air samples?
I try not to. You mean you stick the sampler inside, like the wall check? There's a wall check, and you stick the little sampler inside the wall, and you collect the sample. Now, the problem with that is that one penicillium colony makes maybe 1 million spores or more, and one little penicillium colony. And so you happen to put your sampler right next to one little penicillium colony, and you get 1 million spores on your sample, and then you start tearing down the walls because you've got 1 million spores in that wall, and you have to get them out. Well, you know, 1 million spores inside a wall in one little penicillium colony isn't going to hurt anybody, and it's certainly not going to hurt the wood. It's not going to hurt anybody. And so that's a waste of time. It gives you data that when you sample inside of a wall, you're essentially sampling all the air in the wall. And then, which is a very enclosed space, and it's a place where there's no chance for the air to be mixed with the air that you actually breathe. And so to say that there's that many spores per cubic meter inside of a wall, I think is odd data. It's data that I would hesitate to try to interpret. If you do a wall check and you find a lot of stuff that really looks important, go ahead and open the wall. I don't care if you open walls. I mean, the homeowner might, but as long as they have matching paint, it doesn't really matter. You can fix it so that it's perfect. But I have trouble with wall checks just because the sample in such a confined space, and the molds are tricky.
Okay. Next question: given the high variability of fungal air sample data, is there any point trying to apply rigorous statistics?
Not unless you have 2,000 samples. I mean, no. In any one investigation, there's no point in applying rigorous statistics. There is none whatsoever. If you have a sample, I was associated with a large study in a hotel where more than 1 million samples had been collected. Unwisely, but they were collected anyway. And I could have probably taken that data and done some statistical analysis with it, but I didn't think it was worth it. I don't think it was worth it. They wouldn't give me the data anyway. The lawyers wouldn't let me have the data, so it didn't matter anyway. Yeah. No, you can't do statistical analysis on a single investigation. I sometimes do statistical analysis on all the, we have 28,000 culture plate samples from indoors. And I can do statistical analysis on 28,000 samples, but not on 4. In fact, let me just say one more thing. That was the problem with the Stachybotrys thing that a number of years ago, some children developed hemosiderosis, bleeding lungs, and it was blamed on Stachybotrys in the houses of these children. And the investigator did a statistical analysis with like five samples or 5 sites of saying, yes, this baby has it. And it was just ridiculous. The numbers were so low. You just can't do statistics that way. And, you know, he was biased, or that being the answer, because once the Stachybotrys is seen and the babies are seen, it's difficult not to be biased. Subsequent studies for outbreaks, similar kinds of outbreaks in other places, have documented that Stachybotrys is not involved at all. And so anyway, that's the problem with doing statistics on a small number of anything. Statistics are not designed for that.
Great. Next question. Where should outdoor air sample locations be collected? Should multiple outdoor locations be sampled for non-resident buildings?
For non-resident buildings, yeah, I mean, if you have a large building, I would do several samples. I would do a rooftop sample where the air handling units are, wherever the air handling units are, and I would do samples outside of the entry doors. Those are the two places that I would think would be really important, just outside the entry doors. Because what you're trying to do with an outdoor sample is document what's going on in the outdoor air, and then you're going to try to interpret that with respect to what's going on in the indoor air. And so you need to figure out what the aerosol is near where the outdoor air enters.
Okay. Next question. When sampling on a ship at sea, are there any special considerations?
No, well, other than the fact that the samples, there are marine fungi that if they get into a place where the surface is consistently damp, they will grow. Whether they cause any problem, I don't know. I really don't know enough about the marine fungi. But this one that I found, it was called Sterigmatomyces. It was inhabiting an extensive air handling unit that was right by the ocean, right? So all the air coming in was coming right off the ocean and into that. It was a warehouse, a big manufacturing place. So yeah, I think that you need to maybe have the samples. Well, you're not going to find much out in the ocean. The air outside isn't covered with fungi as much as it is other places. But you can still do outdoor air sampling. You still can. If it's windy, you need to be sure you're orienting the sample parallel into the wind. But other than that, and you can do indoor sampling, and in the same way, if you see mold, you might sample it and send the tape sample in to see what kind of mold it is. So in that sense, it's exactly the same. In the sense of what fungi are likely to be there, it may be very different. It may be very different just because of the salt. There's a lot of salt out there in the ocean. And it will influence the kinds of fungi that grow if there's a persistently wet surface. I mean, Cladosporium is always going to grow, but there are other things that could grow as well that we might not notice because we might not be able to identify them on a spore trap, for example. You might end up having to do certainly tape samples if you see anything, and even possibly cultural samples.
Okay, great. Next question. If you cannot rely on outdoor and indoor comparisons, what do you base the mold growth or baseline from?
I don't. Each individual study is unique in some way. And you get a routine going and you do it, but there's no way that you could just go into every single house or every single building and use the same strategy and expect to be able to interpret the data. That's why I say you have to develop a hypothesis in advance, and you have to decide how you're going to interpret the data you get. Are you going to be able to say yes or no? Are you going to have to say this is more than that for some reason? Which is very difficult. You need to be able to say, this is what I'm going to do. These are the fungi that are present, and they shouldn't be here, or this is the wood-rotting fungus, and it's rotting the house, and you need to get rid of it, and so forth. You're not going to talk about health effects. You don't care about health effects. You're not able to do health effects studies. Most of you, I don't know who all is out there, but I bet that there aren't any epidemiological mycologists out there. You do what I said. You do really, really, really good visual exams. You do focused sampling with very specific answers in mind, what you want in mind. Is this a fungus or isn't it? Is this fungus getting off the wall into the air or is it not? And if you want to know whether the indoor air is better than the outdoor air, go ahead and do it. Go ahead and do it. It's less, you know, if you have more spores indoors than outdoors, yeah, you can say that. You can say there are more. They're the same kind of spores, but there might be more. Again, using the mold score, one of those other things. If all they want to know is are they being exposed to more or less mold indoors than out, yeah, you can do the. I wouldn't just do one. I mean, that's kind of no data, but you can do several outdoors and several indoors. You have to do the same volume in both of them and analyze them, and you can say, yeah, your area here is better than the outdoor air, or your area here isn't better than the outdoor air, and we better look for mold growth. Now, that's the thing. If you want to do indoor-outdoor sampling, it has to come in advance. You have to say, okay, I'm going to do this. I'm going to look at the mold score, and I'm going to go in and do my investigation. I think that's a little backwards, because if you go in and do your investigation first, you may not have to sample at all. That's the bottom line. So I think that the whole process I've talked about is the way to go about this. And if indoor and outdoor sampling helps you test your hypotheses, yeah, by all means do it. And I recommend using the mold score, but if you have a better way of doing it, by all means, be able to defend it.
Okay. Next question. When sampling, why does the IAQP go into the building before taking the outdoor air sample?
IAQP, industrial hygienist quality.
You said IAQP.
I don't know what that is, but
okay.
I don't know what that is, but no, say again what the whole question was.
They were saying, where was it now? When sampling, why does the IAQP go into the building before taking the outdoor air sample?
Yeah, I don't. I mean, I wouldn't, but I would do all my sampling outside first. The problem is, there's a couple of problems. You remember that this is another reason why air sampling is very difficult to do. When you're walking around outside, if you were out walking around outside today here in sunny California, you will get mold spores on your clothing, mold spores and pollen and Lord only knows what else on your clothing. And then you're going to go into the building and you're going to shed those spores in the building, and you're going to need to know how much, because you know, if a person walks into a building, they're taking the outside air with them. That's the bottom line. And if you do that, but it's impossible to sample inside unless you come from outside. You know, it's impossible. So you have to just deal with that. And I think that going inside is important. I think that looking for fungi, looking for water damage, evaluating the whole situation is the first thing you always do, I think. And then at that point, if you think it would be important to get an outdoor air sample because there's some odd things that you see, or you don't see anything and you just want to make sure, then yeah, sure, go ahead and do it. But it's not just an automatic thing that anybody should do. Indoor, outdoor air sampling.
Yeah, I guess they clarified. It stands for Indoor Air Quality Professional. So they're
thank you.
Yeah, so they're saying so I guess I'll read it that way. When sampling, why does the indoor air quality person go into the building before taking the outdoor air sample?
Because he needs he or she needs to do a good in-depth visual inspection, which may solve the problem before you ever need to do any sampling at all. And one of the big wigs at EMLab PK told me that was fine for me to say that. I would say it whether he thought it was fine or not, but he wants our clients to know as much as they possibly can to be doing their work from an intelligent point. A point of high intelligence. That's what I was trying to say. Okay, go ahead.
Great. Next question. Can you explain indoor, outdoor scores and how you came up with your answers?
Well, our indoor-outdoor scores are based on the mold score, and I can't explain the statistic that produces the mold score because I didn't make it. I didn't make it, and I don't know how it was made. I don't know what all the individual steps are. It's a computer program that we put the data into, and we come up with this mold score, and it's based on looking at each individual taxon, each individual kind of mold spore that has been recovered, and comparing indoor and outdoor concentrations by individual mold spore type, and then weighting the spores by the likelihood that they would be growing indoors. And so, for example, bacillospores, it has a relatively low likelihood, and ascospores. So we have really high bacillospores or ascospores indoors. We don't count those as a high indoor count. We count them less stringently than we would if they were penicillium, for example. And this is based on the data that we used to develop this. The programmer just came and talked to each of us all the time. And he would come to our meetings, and we would all sit there. It took us forever to do this. We were traveling around each other's build places to do it. He would come and talk to us about what questions he should ask of the data. And we, as people who've been interpreting data for years and years and years, would say, no, you can't do that because. And so he would fix that part. And then, oh, no, you can't talk about rust because rust never grows indoors. Okay, we'll fix that part. You know, those kinds of things. That's how it was developed. And he kept changing the algorithm until it agreed well with the experts' opinions. And so, as I said earlier, what the mold score is a group of experts, 5 experts, I think, 5 of us. You're getting our opinion on what that data would be, how that data should be interpreted. And it might not be any better than your opinion, and your opinion might be a lot better because you're standing right there in the space that it's happening, but it's what we assume is correct just from doing these for many, 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 many years. That's all I can say.
Okay. Next question. Should we just ignore agreement ratios and Spearman's rank correlations?
Absolutely.
Okay, next question. What is raw count versus spore CT/M3?
Okay, a raw count is how many spores were actually counted on the sample that you're sent. So the analyst sit under the microscope and goes 1, 2, it counts like, let's say, alternaria 1, 2, 3, 4. And then with alternaria, we sample the whole slide. So he'll sample 1, 2, 3, 4, 5, 6, 7, 8, however many there are on the whole entire sample. And then we have to look at how many spores per cubic meter that represents. And the reason we have to do that is because everybody samples, many people sample with different volumes. And so we need to know how much volume of air those 8 spores came from. And so we divide the number, that 8, by the number of liters that were sampled, like 0.75. It's usually 75. It's usually 75 liters that are sampled. And it's if you do that, let me see, you have to divide by, what we're trying to do is divide by the fraction of a cubic meter that were sampled, the fraction of a cubic meter. And that's where that number comes from, that 8. It starts with 8, and then it's transferred into spores per cubic meter by dividing the fraction of dividing that number 8 by the fraction of a cubic meter that was sampled. Now, sometimes there will be a number 8, and then in the next column will be another number that's a little higher, and that means that, well, we sampled half of the sample, or we sampled a third of the sample, in which case we would adjust the counts based on that. So if we sampled half the sample, we would divide the count by 0.5. And if we did a third, we would divide by 0.33. And that gives you a higher number because it extrapolates to the rest of the sample. And the only time we do that extrapolation is when we're at high concentration, high magnifications, where it's physically really almost impossible to count the entire slide. And believe me, there are no laboratories that count the entire slide at high magnification. None of you could not afford the analyst time. Nobody could afford the analyst time to do that. So what they're doing, and we are doing, is looking at the whole slide at low magnification, counting all the things that are big that we can actually count at low magnification, and then going to higher magnification and counting a fraction until we get up to some basic number of spores, and then calculate how much of the sample we've analyzed and adjust the counts by that. And then we take those adjusted counts and divide by the fraction of a cubic meter that was sampled. I hope that worked. I mean, that's right. I know it's right. I just hope it's I was going to say legible, but you know what I mean. Again, if you want that written out, you can email me. Put it in an email question, and I'll write it out for you.
Okay, great. Next question. Can you elaborate on the pros and cons of conducting wall cavity air sampling? Do you recommend it?
I don't recommend wall check, where you just stick a sample in, I don't even know how wall check actually works, but apparently you stick a sampler with a pump on the outside and sample the air inside the wall. And as I said before, you're essentially sampling all of the air in that particular space, because there's not much air in there. There might not be any more air than 75 liters. So you're not only pulling you're pulling all the spores off of all the colonies that might be in there. And if there's one colony, you will get all the spores off of that colony. If you're doing an ambient air sample, you would not get that. You wouldn't be disturbing the colonies. So what you're effectively doing is collecting everything that's in that wall cavity, and then extrapolating from that up to spores per cubic meter of air, which isn't relevant because you've disturbed. It's an aggressive sample by definition. It's an aggressive sample, and it's an aggressive sample that that you can't do the tape sampling to tell whether or not it's actually important. You can't do a tape sample first and say, okay, it's this particular fungus, and now I'm going to look for it in the air. You can't do that with that. So you're just sort of believing. As far as wall sampling, you can do it. You can do it. I just want extreme caution to be applied in interpreting it, because you may be destructing a bunch of walls that you shouldn't. I shouldn't say this, but I will. I like mold dogs because dogs can be trained to sniff out anything. They have dogs that can tell when a child is going to have an epileptic seizure by the scent of that child changing. I mean, that's how good they are. And if a dog is trained on mold volatiles, that dog can tell you when there are mold volatile eggs coming out of a wall that you would never know were there. I have a friend who has mold dogs. I know several people who have mold dogs, actually, and she's never wrong. Her dogs are never wrong. When she opens the wall, there is always mold growing where the dog has pointed. So I'm kind of in favor of mold dogs. It would be nice, and people have tried to develop an electric nose that would do that same thing. It has to be very, 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 very sensitive. Dogs are amazing with how much they can detect. So that's kind of the best. Otherwise, you're going to have to do the wall check, and you're going to have to check for humidity, check for water. How much humidity is there in that air? And if you have no water in that wall space, there might be fungi in there, but they're just sitting there, they're not growing.
Okay,
But
great.
that's all. That's all. Okay.
Sorry, it looks like we have time for maybe one more question. It looks like we've got through most of them anyway. The last question here: do you consider the fact that if the wind is coming from the ocean, outdoor mold is close to zero?
I know for a fact it isn't necessarily, because for one thing, it's clear that spore clouds can be transported clear across oceans. When there are sands, I can't remember where it was reported, but sandstorms and dust storms, those clouds being transported for hundreds of miles in clouds across water to another place. So these are very small particles, and if the wind is blowing, they stay airborne and go for long distances. So in general, you're going to find, if I went over to Point Reyes and stuck out a sampler and sampled air off the Pacific, I'm not going to get much, assuming that the wind is blowing steadily off the Pacific. But there's nothing out there for 2,000 miles that could give you any mold spores, nothing. So you're much on the east coast. It's more likely that you would see long distance transport. There's a lot of literature on long distance transport of fungal spores. Basically, it's in the agricultural literature because they worry about, because they worry about spores, pathogens transporting from one place to another, pathogens that might be controlled here but aren't controlled somewhere else. So we know, and even bacterial diseases, there's a bacterial disease of cattle that has clearly transferred from one island to the next. So I guess that's my answer.
Okay, great. Do you have any final comments, Dr. Birch, before we sign off?
Yeah, I think that if I've confused you, or if you have further questions, or if you want to repeat your questions and send them off to Dave so that I can read them and try to interpret them in a more detailed way, by all means do it. I'll be glad to do it. Otherwise, I've enjoyed talking to you, even though I couldn't see you, and I hope to meet you sometime. Come up and say hello if you're at the AIHC. Thanks.
Great, thanks so much. And as a final reminder to our audience members, a certificate of attendance will be provided to those who complete the post-event survey. We'll post the survey here in the room in just a minute, so stick around for that. We'll also send that same survey in a post-event email in a couple hours from now. It's the same survey, so if you complete it here, no need to complete it again when you get the email. But the deadline, the window to complete the survey is 24 hours from now. That's when the deadline for the survey will conclude tomorrow. So please complete it as soon as possible. Further questions about certifications can be emailed to webinars@emlabpk.com. And again, content-related questions should go to Dave Gallup at dgallup@emlabpk.com. This webinar has now concluded, and you may now disconnect. Have a great day, everyone.